Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not...
Okay.
All right. Good morning to everyone in the room, and good morning, good afternoon, or evening to everyone online, depending on wherever and whenever you may be. Welcome, everyone, to day 2 of our Advancing Generic Drug Development Workshop. We had great sessions yesterday and are so happy to see everyone here back for more. As a reminder, for those on site, in addition to the lectures, we also have some posters for viewing just outside these doors, so be sure to check them out. We're also offering live YouTube streaming, so go ahead and invite your colleagues to view online. However, please note that only those attendees who registered and are attending via the room logins will be able to submit questions to our speakers. And as always, we encourage you to make the most of this experience, attend all the sessions that are of interest to you, learn all that you can, and be sure to ask your questions during the Q&A sessions. For our online logged in attendees, submit your questions using the online link and QR codes. So for those of you who don't know me already, my name is Lieutenant Commander Nora Lim, and I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. And I'll be your host for today's sessions. And now for some of our CE information, we're pleased to announce that this 2-day workshop offers a total of 10.5 continuing education credit hours to physicians, pharmacists, and nurses. The CE are available to all real-time attendees who attend via the online login. Please note the emphasis on real-time and online login. No CEs can be given for those who attend via YouTube or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty or speakers are required to disclose to the attendees when products or procedures being discussed are labeled or off-labeled, unlabeled, or not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers, and the planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete the final activity evaluation via the CE portal. That's ceportal.fda.gov. For multi-day activities, participants must attest to their attendance and complete the faculty evaluations for each day. So final evaluations must be completed within 2 weeks after the activity, no exceptions. So in this case, for this 2-day workshop, the deadline is going to be October 9th. And we apologize in advance, but we're unable to make exceptions to that deadline. Oh, sorry. Pharmacists should note that failure to provide their correct NABP and date of birth information in the required format may result in the loss of credit for the activity. Your NABP profile number should be the 6 to 7 digit number assigned by the CPE monitor, and your birth date should be in the 4-digit month, date format. So for example, like today, that would be 0925. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to the Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print their statement of credit. Pharmacists should log into the CPE monitor 6 weeks after the last session of the activity to obtain or view their CE credit. And we strongly encourage you to complete the evaluations upon receiving our thank you for attending email, as we won't be able to assist you if you miss that deadline again of October 9th. So again, here are our general objectives for the workshop. I'll give you a moment to read through the bullet points. Okay. We'll also take a moment here to highlight our upcoming events. So on October 9th, we'll have a webinar that's the ICH M12 Drug-Drug Interaction Studies Final Guidance. On October 16th, a Global IDMP Implementation, Getting Closer to the Goal webinar. On November 7th, we'll have a Putting the Pieces Together REMS Logic Model in REMS Design, Implementation, and Evaluation webinar. Be sure to visit our website, that's fda.gov/CEDAR-SBIA, and sign up for our listserv to stay informed of our upcoming events. More information is available on our homepage here, and we also invite you to visit our CEDAR Learn webpage, where you can find links to SBIA tutorials, along with other educational resources, many of which offer healthcare professional CEs as well. Okay. All right, for those of you that have attended before, you probably know how this goes, but we'll go ahead and review some logistics as it applies to all our attendees. Here's that link to our one-stop shop webpage for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. For our online attendees, this is the same page that you used this morning to access the event. So if you have technical questions about the conference, please check there first and you'll most likely find the solution. The one-stop shop page also has the evaluation links. It's also where you'll find the final survey, which will be activated this afternoon and will be sent in the post-event thank you email as well. Upon completion of this survey, attendees can download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The trick is that the certificate has to be downloaded immediately after completing that survey, otherwise the opportunity is lost. And even if you do not want or need a certificate, please take a few minutes to complete those evaluations, as your feedback will help guide the design of our future events. And just another note that that deadline date is October 9th. And one more reminder, all the sessions are being recorded. It will take us a couple of weeks to process the recordings, but you will soon be able to access them from SBIA's homepage, and that's fda.gov, CEDAR SBIA. And in the interim, we'll also have the YouTube links for both days that will be posted on that one-stop webpage at the end of the day as well. Also, to simplify things, within 24 hours of the conclusion of today's activity is when SBIA will send one email to attendees of the individual event day, which will have that information on both CE claim code and survey link. And one other reminder that questions can be submitted for our online logged in attendees using that on-screen link and QR code. So here are the logistics again, specifically for our in-person attendees. Here is the on-site Wi-Fi information. And for Q&A, we're going to ask that you please line up at either of the mics during the Q&A panels. We have one on each side of the room there. Line up to ask your questions. Another reminder to please silence your phones. Use the internet on-site sparingly, as it may negatively affect our online delivery. And sorry, but you're on your own for lunch today. We do have flyers at the registration desk, which you can use to scan QR codes to pre-order for nearby eating establishments, if that helps. Right. One more thing, we'd really like you to help us help you to stay informed of our events. We're trying to determine the most effective method for advertising our SBIA events, and so we need your help, both for online and in-person attendees. We are asking that you please take our survey and let you know what you think. Flyers are available at tables and at our SBIA exhibit table. For online attendees, you can also scan the QR code and respond accordingly as well. All right, so you may already know Captain Brenda Stoddard and myself as your SBIA hosts, but I'd also like to give special recognition and introduction to my colleague, not pictured here, but here with us today in the room, Dr. Corey Adair, a fellow pharmacist with the SBIA team, who will be assisting with the readout of our online questions submitted during today's Q&A panels. All right. And now, the moment that you've all been waiting for, it is time for us to launch fully into today's agenda. Note that speaker bios are available online on our one-stop shop webpage. It's now my pleasure to introduce our speakers for today's first sessions, which will be a spotlight on generic drug review challenges and solutions. First, we'll hear from Tina Zhao, a chemist with the Division of Product Quality Assessment for Office of Product Quality Assessment 1 within the Office of Pharmaceutical Quality. Then we'll hear from Dr. Maria Flynn, a Senior Pharmaceutical Quality Assessor within the Division of Product Quality Assessment 8, also within the Office of Product Quality Assessment 2 in the Office of Pharmaceutical Quality, and followed by a presentation by Dr. Nashwa El-Gendi, Senior Pharmaceutical Quality Assessor within DPQA5, and again, the Office of Product Quality Assessment 1 within the Office of Pharmaceutical Quality. All right, and now I'll hand it over to Tina Zhao.
Okay, today I'm going to talk about quality-related review considerations in teriparatide injection first generic approval. So my presentation will cover quality-related recommendations and the immunogenicity risk mitigation for teriparatide injection generic applications. I will also go over some examples of common deficiencies for NA peptide products. My presentation will help you to learn quality-related recommendations for teriparatide injection generic approval, and also some common deficiencies to highlight aspects where attention should be focused to facilitate ANDA approval. Teriparatide contains 34 amino acids, is indicated for osteoporosis. The drug product is a clear solution in a multi-dose injector pen for sub-Q injection. The reference product is from Eli Lilly with recombinant API. The first approved generic ANDAs are from Teva and Apotex. Both are Q1, Q2 the same as the RLD, and both are eligible for biowaiver, and both with the synthetic API. For ANDA submitted under 505J, the applicant must demonstrate that the proposed generic drug has therapeutic equivalence with the RLD. A drug that is therapeutically equivalent also needs to be pharmaceutically equivalent and bioequivalent. I want to point out the same active ingredient is required by both PE and BE. For teriparatide injection, FDA just published the product-specific guidance in this August. Besides the Q1, Q2 and the device recommendation, this PSG also direct the applicant to follow this FDA peptide guidance for the quality considerations. In my slides, I also list those peptide products that whose PSG also reference this peptide guidance. I also list those peptide products whose PSG recommend active ingredient sameness. The FDA peptide guidance describe the comparative characterization of the following attributes for the proposed drug product and RLD. Primary sequence and physical chemical properties, secondary structure, oligomer aggregation states, biological activities, and also the use of orthogonal analytical methods is recommended. These slides provide more detail of the active ingredient characterization and comparability studies to characterize the drug substance primary structure and physical chemical properties. The firm needs to provide evidence to ensure the identity, strength, quality, and the purity of the peptide drug substance. DMF can be used for this AI characterization. For the comparability, the firm needs to provide evidence to ensure the peptide higher order structure, oligomer aggregation profile, and the biological activity in the proposed finished drug product are comparable to those of the RLD. This slide provides the examples of characterization comparability studies at the drug substance level, and also some examples of the analytical methods. To characterize the primary sequence and physical chemical properties, the firm's study needs to include primary peptide structure sequence, solubility as a function of pH, high growth capacity, log P, isoelectric point, specific optical rotation. The analytical method could be IR, MS, UV, NMR, CD, AAA, L and D amino acid content, AAS, Edman degradation, and or high resolution MS. These slides provide the examples of characterization comparability studies at the drug product level and some examples of those analytical methods. For the higher order structure, the far UV-CD, FTIR, Raman can be used to characterize the secondary structure. Near UV-CD, intrinsic fluorescence, DSC NMR can be used to characterize the tertiary structure. For the oligomer aggregation states, the light scattering, analytical ultracentrifugation, gel electrophoresis, FFF, MFI can be used to characterize reversible non-covalent soluble oligomers, and also those irreversible soluble insoluble aggregates. For the biological activities, bioidentity tests per USP monograph for teriparatide injection can be used in the therapeutically relevant bioassay. Moving on to the impurity comparability studies. Per the FDA peptide guidance, the impurity should not be greater in the test product than that in the RLD. The firm needs to conduct a comparative peptide-related impurity profiling of the RLD and the proposed generic product to identify each peptide-related impurity at level greater than or equal to 0.10%, to demonstrate the no new peptide-related impurity greater than 0.5%. For impurity found in both the proposed drug product and the RLD, the firm needs to demonstrate the level in the test product is same as or lower than that of the RLD. For impurity found at level between 0.10 and 0.5%, if not present in the RLD or in the test product at higher level than that of the RLD, the firm needs to provide justification as to why such an impurity does not affect the safety, effectiveness, or potential for immunogenicity. And also the firm should consider to use the UHPLC high-resolution MS to facilitate peak identification and ensure peak purity. Regarding the immunogenicity risk mitigation, for any new or elevated level impurities found at level of between 0.10 and 0.5%, adaptive immunogenicity risks for such impurities should be evaluated individually on the purified impurity standard. Innate immune response evaluation should be conducted on the fully formulated drug product in comparison with the ROD to ensure the generic product does not contain anything such as impurities, aggregates, contaminants, or leachables that may increase immunogenicity risk. Next, I want to go over some common deficiencies for any peptide products. The first example, the firm performed an impurity profile characterization of the test product samples aged near release using the HPLC-related substance method. FDA recommend the firm to update the study using test product samples aged at the end of the proposed shelf life and perform the characterization study using orthogonal methods that use different chromatographic separation principles and also use UHPLC high-resolution MS to facilitate peak identification and matching between the ROD and the proposed the product to characterize peak purity. The next example, the firm identified some impurities in the spec table with RRTs. FDA recommend the firm to characterize the structure of those impurities identified by RRTs as per the ANDA peptide guidance and update the spec table with identified name based on the characterization. The next example, the firm proposed a limit of no more than 0.10% for any unspecified impurity. FDA recommend the firm to revise the limit from no more than 0.10% to less than 0.10% to be consistent with the ANDA peptide guidance, which states that all impurities presented at 0.10% or greater should be identified and characterized. The next example, the limit proposed by the firm for impurity 1 is more relaxed than no more than 0.5% and not supported by the comparative RD data. FDA recommend the firm to tighten the limit consistent with and supported by the observed the RD level, or tighten the limit to 0.10 to 0.5% and supported by immunogenicity study results, suggesting the impurity 1 does not add immunogenicity risk to the product at the proposed limit. The next example, the firm reported a greater level of aggregates in the test product than those found in the ROD. The FDA recommend the firm to perform an investigation to identify the root cause and improve the product aggregation profile. The aggregation profile of the test product should be comparable to the ROD. And also the firm needs to demonstrate the capability of manufacturing drug substance or drug product, exhibit batch as necessary with acceptable control of aggregates across the proposed shelf life. Now we're moving on to the challenge question. The question number 1, consistent with the ANDA peptide guidance, when conducting a comparative impurity profiling of the generic product and RLD, identify all peptide-related impurity at a level of A greater than 0.10%, B greater than 0.5%, C greater than or equal to 0.10%, D greater than or equal to 0.1%. The answer is C. Per the FDA peptide guidance, the firm needs to identify all peptide-related impurity at a level of greater than or equal to 0.10%. Challenge question number 2. Which of the following statements is not true? A, ICH Q3A and Q3B guideline is generally applicable to all drug products, including peptide drug products. B, the limit for each peptide-related impurity can be qualified by the observed RLD level for the same impurity. C, a comparative characterization of the impurity profile of the generic product and RLD is recommended to establish acceptance impurity limits. D, peptide-related impurity and aggregates may pose immunogenicity risks. The answer is A. Per the ICH Q3A and Q3B, the peptide drug product are not in the scope of ICH Q3A and Q3B. In summary, my presentation provided the overview of the quality-related recommendations relevant to teriparatide injection generic approval, and also provide some, we go over some examples of common deficiencies for those, for any peptide products. Lastly, I want to thank Cameron, Ely, and Bing for their help on my presentation. That's it. And next, please welcome Dr. Flynn.
Good morning. I'm Maria Flynn, and I have a question for you. Do you know someone who has not been able to afford their prescribed medications? You likely do, even if you don't know, because 1 out of every 3 Americans can't afford their medicines. In fact, if they could afford them, they may not be able to afford the most advanced forms. First generic drug approvals bring down the cost of medications. Today, I'm going to be referring to the first generic oral liquids, and those approvals actually done just recently. Oral liquids are very important to vulnerable populations, such as our geriatric populations, our elderly or wise old friends, as well as the youngest, the pediatric populations, and other patients who have swallowability issues. So today, we're going to have oh, there we go. He told me to look panic. So our learning objectives today is we're going to discuss the quality attributes for oral liquids. And we're going to do this in the context and the framework of 2023 approved applications. And so we're going to be discussing 4 case studies. The first 2 case studies, we're going to be discussing the stability of an active pharmaceutical ingredient, also known as an API, or actives. And so the very first case scenario, we're going to be discussing physical stability. And when we think about physical stability, we're actually thinking about the structure of the active. In the second case scenario, we're going to be talking about the chemical reactivity of that active. So if it can degrade, something like that. And in the third case, we're going to be discussing the unexpected leachable situation, as well as our final, which is going to be a container closure risk management scenario. So this slide is showing all the possibles, well, not all, some of the critical quality attributes. You always have to leave room for that. And moving forward, I'm going to be discussing very specifically in the case studies, I'm going to be specifying if it's an oral solution or oral suspension. So when you think about oral liquids, I want to refer you to a chapter in USP. And there's a chapter 1151 that goes through all the different dosage forms for oral liquids. And in that, they'll define each one. So I will not be discussing today the other dosage forms, such as a powder for solution or suspension or syrups, because the last first approvals from 2023 were oral solutions and oral suspensions. So in thinking about the critical quality attributes, we already discussed stability. And why is that so? So when you're in a liquid phase, unlike your tablet where you have your active that's in a fixed position, you have fluidity. You can actually see many things because you're a liquid. So if I were an active, I could see other actives. I can also see other excipients. I could see solvent and all the permeations above. So because of that, they are more reactive. And a lot of times, you will see that you have to have different storage conditions. They might have shorter shelf lives, and they might have some instability. And so we talked about physical stability and chemical stability. And we were looking at assay. We assay the active. And sometimes the active, you can actually see if it is degrading, you can see changes in the assay. But there are also other functional parts of the formulation that we are going to assay as well. Content and uniformity is applicable, but I want to even throw out there another term for oral suspensions, and that's going to be dose uniformity. And so this is very important when you think about suspensions. All throughout the bottle, a lot of the cases, they'll tell you to shake it. And all throughout, you want to make sure from the top, middle, and bottom that you get a homogeneous dose given to the patient. And also over its shelf life, you want to look at dose uniformity. Container closure systems are very important for liquids because they have to be airtight. You don't want to have leakage or spillage. Dosing accuracy is critical in making sure you get the right dose to the right patient for the right indication. Microbial limits. You're in a liquid, so there's water activity that can promote microbial growth. So microbial contamination as well as controls need to be in place. And a lot of times we'll see preservatives being used to be antimicrobial. Leachables are relative to our solid oral dosage forms. They're of higher risk. However, maybe not as much risk as we saw in TNUS when you're dealing with injectables. Dissolution, we're going to reserve to oral suspensions because the active's being suspended, and so you care about it being into how it goes into solution. So let's go ahead and dive into our first case study. When we're thinking about physical stability of oral solutions and suspensions, we're thinking about the solid state. And this is for both dosage forms. You're thinking about the different forms that can occur. The last two, sedimentation and particle size growth, this is only reserved for oral suspensions in that that's what, when you have to shake it, it can either sediment or there could be particle size growth over time. And you need to control that again for dose uniformity. So those we'll reserve. But I want to also throw out there another comment about redispersibility and resuspendability throughout the shelf life in each dose. So with physical stability of oral solution and liquids, we think about solid state, and we can ask ourselves some questions, for instance, about the active. Is it known to have polymorphism? If it does, there might be an increased risk. We can ask, is the form crystalline? Is it amorphous? Suspensions can have a much greater risk than solutions because in suspension, it's thermodynamically unstable. So inherently, it can have more risk. But whenever you're thinking about different forms, you really need to think about the solution equilibrium of each form. So when we're thinking about solid states of the oral solution, you can think of it lower risk because it's all dissolved if it's a true solution. And if you know that your active is dissolved in a concentration that's less than the solution equilibrium, you can think about a lower risk. Now, for oral suspensions, while we mentioned that they're at higher risk, there's some things that you can think of initially that you can think that it may not have as much risk. For instance, if there's no polymorphisms, it's not existing, you don't see it, then, and it's not in the literature, and so far under your control, you don't see it, it may not be a risk. Or you're using the most stable form. And then there's times when all you know is the amorphous form, and in that case, there's a likelihood that there wouldn't be a crystalline form. So that can initially lower your risk. But when we also think about actives, we think about them in terms of solubility. And with solubility, we think about the high solubility class, and we use a class system called BCS, which is the Biopharmaceutical Classification System, and it explains what would be considered highly soluble. So if your active is highly soluble, it's more likely it's not going to have risk. Now, you can increase the risk if you know that you're using the unstable form. And if you also have multiple forms and you know they interconvert, that can also increase your risk. So let's dive into our first case study, and we're dealing with an oral suspension. And this oral suspension is known to have polymorphism. You can see the different forms. It's crystalline, but again, as I mentioned, the active is being suspended, okay? And so there's a greater risk thermodynamically for that. It happens to also be in a risk category of poor solubility, a BCS class 2. So what do they do? What do we do in this situation? Well, you need to mitigate it. Before you even go into formulation and development, all that wonderful things, you need to control your drug substance. You want to make sure you know the polymorphic identification. You want to have a 3-tier particle size for the drug substance. This is before you even get to the drug product. Then, once you know that, you go through manufacturing. Things can happen during manufacturing. In the very end, you get your drug product. So at release, you want to make sure that that polymorphism is what you have. It has not changed. But also, over the shelf life, you want to make sure that it remains in that form. Now, in the drug product also, you want to make sure you have a 3-tier particle size for the actual suspension, that you know what exactly the particle size, and there's a range, and there's 3 tiers. Now, I show you a number that's less than 22 microns, but there are 3 different numbers that make that up. I wanted to put that number there specifically because I wanted to show that this is highly micronized. And this is a trick that pharmaceutical scientists do. We love to, I don't know if I can call myself a pharmaceutical scientist, but I'll put myself in the category since I have 16 years experience. We love to have very highly micronized compounds because you increase the surface area, which then increases your dissolution rate. For pharmaceutical, so that's your going into and what you know. But furthermore, since we knew that this fit into such a high risk category, the applicant for this first generic showed a polymorphic study. And they showed that at the 6-month accelerated, as well as at the 24-month, the long term, which happened to be the shelf life for their drug product, there was no conversion. So they had a good control, and they're going to keep all these controls in place and watch it, and so make sure that this does not happen and that they have the form, because it's very important. So now diving into chemical stability, we're going to be talking about different risk. Usually by the time an applicant comes to FDA, they have at least 3 months, usually 6 months of accelerated as well as long-term data. And at that first initial risk, you can either see that there's no trend, or you can see significant trend. If you don't see trend, initially, it might have a low risk. That doesn't necessarily mean that will happen over the course of the shelf life, but you can feel a little bit sense of security. When it comes to risk, when you see significant trend, so if you see the assay decreasing, for instance, or you see other degradants forming over time, these significant trends, and especially if they go out of spec, really give you a heads up that there's something unstable about that formulation. It could be obviously we're looking at the active in this particular situation. But there's also tricks that we do in pharmaceutical science that actually stabilize. And so if you see stabilizing agents, for instance, specifically for the API, if you see that going on, then it might give you a heads up that there's a higher risk. So this case study is talking about the API degrading. Even though we're talking about oral suspension here, I can't tell you how many times this come across our desk, where this is universal to all dosage forms. So I put the label here specifically. Sometimes you can get some hints in the RLD labeling about what possibly can go awry, or what can be the failure modes for your formulation or their formulation. So in this particular case, with much evidence I'm not going to show here, the API was thermally unstable, so it was affected by heat. So when you look at the stability conditions, how they knew that this was happening in their specific situation is that they saw at the 6-month accelerated, and I want to mention here, this is a refrigerated suspension. And so the accelerated is at 25 degrees. And they actually saw a significant trend in the assay. And when we say significant, it means greater than 5%. And they also saw out-of-spec degradants. In the long term, the refrigerated, everything was fine. It complied with all the criteria. So the problem is, if you read our guidance the guidance for ICH-Q1E, for refrigerated compounds, it's very important that it be supported by real-life data. And so in this case, they wanted 24 months stability or shelf life. They wanted that. And so they needed to back that up with real science. And there's a little bit more going on for the suspension. And so it was very important to see that they had a stable formulation, formula. So we're moving on to our first challenge question. And I want to, sometimes we read these quickly, I want to put your eye in the lens of the API. What is not an active pharmaceutical API-related risk factor for oral liquids? So A, exhibits polymorphism, B, dosing cup inaccuracy, C, poor solubility, D, easily oxidized, air sensitive. So I'm going to give you a couple seconds to think about that. So the answer is B. Dosing cup inaccuracy is a risk. It's just not related to the API. The other three can be failure modes for actives. So moving on, we're going to discuss a case 3 study where you're seeing an out-of-spec degradant. This one's very interesting because only one batch of the 3 exhibit batches failed at 6 months accelerated and at the 24-month long term. In fact, the degradant that they didn't even know they had popped up and started to exceed the limits of ICH-Q3B. I want to refer back to Tina. She had a special case when it came to peptides. They had a much stringent, in this particular situation, they didn't have anything very special about their active, and there was no cohort of concern, but they were able to use the guidance ICH-Q3B for their drug product. So this degradant it actually exceeded the identification threshold criteria. So under the identification threshold, you don't really need to know, you might want to know, but you don't really need to know what is there because it's considered safe. So what's going on here? They had to figure out what this was because they now need to find out why this one batch had this degradant. And so they identified and characterized, and imagine, this is a first generic approval, so there's no standards. So they needed to confirm this. They do all the wonderful organic chemistry, and they do I'm actually an organic chemist, and so they actually did the structural elucidification, but to really confirm, confirmation of this is the synthetic, actually synthesizing this. So what did they have? Well, they found out it was very similar to an oxidative degradant they already knew about, but it was a diastereomer, which means one of the chiral centers was racemide. And thinking as an organic chemist about how this can happen, it wasn't just like an acid, like the pH of the oral liquid or suspension. Oh, this is actually an oral solution. In that situation, they couldn't really think about how this could really happen. So they had to think a little more. Why did that one batch out of the 3 fail? What happened? What changed? What was different? So they did their homework. What they learned was that one batch, after much thinking, that they were using one normal HCl to adjust at the very end that drug product formulation. And the difference there was the one normal HCl sat in a stainless steel vessel for longer than the other 2 batches. They found that there was iron being leached out of the stainless steel vessel into this acid that is now going to be used for the drug product formulation. And an oxidized reaction catalyzed by the iron can make that degradant. And they went along and did the science and showed that they did have iron that was a higher concentration than the other ones. And they also realistically, theoretically, it made sense. So that explained why this degradant was growing over time and that why they failed stability. So with that in mind, they had a control strategy. They revised their specifications, and they now are controlling this degradant, and they controlled it at the ICHQ3B. Again, they're allowed to do that. And they committed to even further to do this, since now they understood it, that they would go ahead and they would control this at even a lower limit on commercialization. So what did they do well? They submitted a well-written CAPA, a corrective action preventative action report. They understood the origin of the degradant. They confirmed it by synthesis, and they used a robust analytical methodology to actually see it. And not only that, they committed and they will specify the degradant at a tighter limit, not that we had asked them to do that. They chose to do that. And they got first generic approval. So here's our second question, and I know we're short on time, so I'm going to move quickly through this. All are common deficiencies for oral liquids. So what is a common deficiency? Would it be a well-written CAPA report with a control strategy for unexpected finding? Is there an incomplete understanding of the manufacturing process? Is there not specifying the container closure description and integrity? So if you think about this, what is a common deficiency? You know the last 3 are common deficiencies, but a well-written CAPA and understanding an unexpected finding, that's a good thing. So all the other ones would be common deficiencies. So moving right on, now you reversed you're doing a first generic and you reverse the RLD, reverse engineering, you know what's going on, everything's so wonderful. You made this wonderful formulation, and then you choose the wrong container closure system. So we want you always to look at the packaging evaluation and make sure that even over the shelf life that you are not having any problems with your different configurations. Because in our first case, and our last case, we're going to be discussing this particular picture here, which is 1,000 words. This seal was not appropriate for this single-use unit. And what happened, this is very bad for the patient, they're not going to get the right amount of dose. But furthermore, this could be a hazard for the healthcare professionals and just a mess, depending on what the drug product is. So this was withdrawn. And this first generic approval for the multi-dose containers that had a good seal, they were fine, and they were able to get first generic approval. However, this one is not acceptable. And this is inverted, so when we do these liquid studies, we do the upright, and then we do the worst case. So it could be horizontal or it could be inverted. In this particular case with the single unit dose, it was inverted. So in sum, we discussed the quality attributes and the risk of oral suspensions and liquids. We also discussed risk mitigation possibilities and how they were actually done for 4 case studies in real life for first generic approvals. So I have some closing thoughts. Know your drug product, understand it. Think about the failure modes and the risk. Come up with a mitigation strategy. But when all that fails, expect the unexpected, because the unknown unknowns will occur. But when they do occur, really find the root cause analysis, really do the kappa, and submit that with a very clear control strategy of how you're going to go forward so that you can get a first generic approval. So now I'd like to welcome up Nashwa.
Good morning, everyone. My name is Nashwa Al-Gindi, and my presentation topic today is about quality consideration for first generic tiotropium bromide capsule-based dry powder inhalers. This presentation will deliver several learning objectives. We will explore the general constraints in dry powder inhalation, formulation, and manufacturing. We'll discuss potential development challenges for capsule-based dry powder inhalers to improve the quality and the performance of these products. Then I will share valuable recommendations for highlighted product characterization studies, and we'll end it with focusing on general quality considerations for primary drug product stability batches. First, I would like to give you a brief introductory about DPIs. There is a lot of many classification for DPIs. However, here I will focus on one classification based on the device constituent part, the dose container. DPIs can be meter dose inhaler, which a powder reservoir contain multiple units in one container, or pre-meter dose inhalers, which are plasters, discus, cartridge that contain multi-unit dose inhalers, or capsule, which is single unit dose inhaler, and this is the focus for our talk. As a fact, generic DPI can be more complex to review compared to meter dose inhalers. Also, plasters and capsule DPIs-based DPIs are the main type submitted to FDA since 2015. So the reference listed product, the RLD for the first generic capsule-based inhaler is Priva Inhaler, NDA 021395 by Boehringer Pharmaceuticals that has been approved in 2004. The first generic Spriva inhaler is Loop Inhaler by Lupin and 211287 that has been approved in 2023. Each capsule of this drug product contains a dry powder consisting of 18 microgram of the API, tiotropium, blended with the carrier lactose monohydrate. There is the product-specific guidance, draft product-specific guidance with this drug product has been published in 2023 and includes many recommendations for BE studies. One of these recommendations is that the test and the reference product recommended to be Q1, Q2 the same. Also, the test and the reference product, the device should be similar in the mechanism, the breath-actuated pre-meter single unit dose capsule-based format. Also, the number of the doses, the size and the shape and the resistance, the external operating principles and the critical design attributes. There are in vivo PK and comparative clinical endpoints recommendation in this PSG. However, I will focus my talk here about detailing about the in vitro BE studies. For the in vitro BE studies should be conducted on 3 exhibit batches, 10 units per batch for the test and reference product. And just here the table summarizes the 2 in vitro studies for in the PSG for this drug product, the single actuation content with the SEC and the aerodynamic particle size distribution APSD. And here in the second column, the design for each study and the summary of the BE analysis. Here they are very related to the quality tests, the SEC, similar to the dose content, delivered dose uniformity and APSD from quality perspective. Then I will jump here into the composition of the capsule-based DPIs. So usually the composition table for sure is usually submitted in the module 3 in P1, which contain the ingredient, quality standard, function, quality, quantity, milligram per unit, and the concentration percent weight per weight. Firstly, the API here, the quantity should be per the RLD labeling, and this is the DPI strength, which is the amount of the amount of the drug from the capsule, not the target delivered dose, which is the mass of the drug delivered from the mouthpiece. Then the carrier, and it should be here based on the PSG for this specific drug, it should be Q1, Q2 the same, and this is the most preferred for most complex product. Then the capsule size and composition, and the capsule shell material may be in some cases different between the RLD and the test, and it may be acceptable or may be adequate in some case. Here in our situation, the listed product is gelatin material, however, the first approved is HPMC material. They are different in moisture content. However, it could be acceptable to use different material, provided adequate justification with supporting data. For example, study to demonstrate that the fragments generated by piercing and actuation of the rupturing of the capsule are not inhaled by the patient. Also, I'm sorry, here also the ink composition should be listed in the composition table. Then I will start with one of the first topic from my presentation about the learning objectives, which is the general limitations for DPIs in general. There is a lot of constraints. Firstly, when we look at the formulation design, as we said before, Q1 the same. Q2, this is preferred. For this specific drug product, yes, it's in the PSG highly recommended. However, for some applicants, they may come up with different Q2. In this case, rationale and adequate justification should be provided, as well as pharmaceutical development data for in vitro testing or studies for a combination of the between different ratios of the drug to excipient, which including combination above and below these ratios for the RLD and test. Justification is really important here, and data. Also, average. Generally, it discouraged per ICHQ8 guidance. However, if it's used, justification should be also provided. For example, drug loss during proposed manufacturing process. Another thing is the manufacturability, and for sure, this is powder. Powder characteristics are very challenging. So many powder should be tolerated, should have a tolerated particle engineering. The shape and the surface characteristics of the powder can affect the aerosol performance and the airflow, and as well as the drug release. Another constraint for the DPI, specifically for the capsule base, is the device. And as I said, per the PSG, the device design is very important to be similar with the RLD design. Also equivalent design, also the operating principles and the resistance. This is very critical attribute for the device. Now, my first challenging question, to keep you in the loop, which of the following examples can justify the proposed average in DPI? A, drug loss during proposed manufacturing process, drug loss due to degradation of the drug substance, drug loss due to deposition on the device constituent part through unit life. The answer is A. Now I'll go deeper into the challenging in developing generic DPIs. To develop a generic DPI with a consistent DPI performance, which represented by the delivered dose uniformity, for example, and APSD. A lot of unique challenges are related to the drug product formulation, API and excipient, manufacturing process, device design characteristic, and ended with the user interface considerations, which is the patient handling. Now, a couple of examples about the formulation development challenges. Firstly, the risk may come from recrystallization of the micronized material, which could lead to particle growth, uncontrolled particle growth during shelf life of the product, thereby affecting the product quality attributes, specifically APSD and DDU. And APSD and DDU are considered the key performance characteristics and measures in assessing BE studies. And accordingly, it also in turn may affect the in vivo performance of the drug product. Also, missing proper control on critical material attributes of the drug constituent parts of this combination product, like the API or the carriers, without proper justification. One of the big challenge here is the amorphous content of micronized particles. Okay, mitigation. What is the control strategy to mitigate this risk? Include equilibration period or condition to allow the recrystallization to occur prior to any additional manufacturing unit operations. Also, consistency of the manufacturing process. Also, establish particle size distribution specification based on the actual data from the micronized lots used in the manufacturing of the drug product exhibit batch or primary stability batches, and ended with continuous in-use control, which is the routine control of the critical material attributes. Another risk here, based on the diagram I can show you here, this is, sorry. So based on this diagram, you can see the powders, the DPIs consisting of API particles, very small, usually micronized within 5 micron, attach it to the surface of the carrier to form agglomerates. This agglomerate fluidize to form a separate agglomerates once it's subjected to the airflow with the passive DPI, with the inhalation by the patient. It starts to be de-agglomerated to disperse the API particles out of the surface of the carrier to be inhaled into the site of action. So the carrier here is very critical component. So the carrier grade may lead to particle size growth over time during storage. Also, the carrier morphology, shape, and surface roughness may affect the adhesion force between the API particles and the carrier particles and affect the aerosol performance. Mitigation control strategy here is to adjust the ratio of the fine to coarse grade particles if they are there, because if the fine particles can be attached to the coarse particles and may affect the attachment or the adhesion of the API particles. Also, continuous in-house control for particle size and morphology. I will jump here into manufacturing challenges. After formulation, we go into the another challenge with the manufacturing, and I will focus in 2 challenges. Firstly, the blend uniformity. Blend uniformity, because this drug product, the challenge here is the excipient over 90% of the whole formulation. So there is a low drug load. Also the flowability due to excipient lot-to-lot variability. How to mitigate these risks with the control strategy? Firstly, how to select the proper blender type, the equipment used. Control the critical process parameters like the blending time, speed, blender fill level. Also the excipient critical material attribute. Control the environmental conditions, conditioning, hold, equilibration period after blending. In addition to the final blend characterization, which includes the uniformity, bulk density, and particle size. Powder fill, another challenging during manufacturing. Fill weight variation and segregation of the API and the excipient lead to failure in the APSD, DDU, and content uniformity. Also, the moisture content, very critical for affecting the APSD and DDU. Mitigation control risk here. It's important to identify the critical process parameters and characterization of the fill weight, content uniformity, moisture, APSD, DDU, and this is from the filled units through stratified sampling during development. Highly recommended. My last challenge here about the container cluster, which is the big deal for combination products. In this specific product, capsule-based product, it has the device, it has the capsule that containing the powder, and the plasters, which is the primary packaging. Here I will give you some recommendations important for this device combination. Firstly, for the capsule dimension is critical for the adequate insertion of the capsule into the inhalation device, piercing, and drug delivery. So it's recommended to include the capsule dimension in the drug product specification, which is the lock length. What about the blister foil and the overwrap pouch of the device? They provide the protection from moisture permeation to the blend formulation at use and in use and shelf life. So it's recommended that the functional layers of the foil should be controlled for the thickness of the individual functional layer, that to assure the consistency, the consistent quality and performance of the primary and secondary packaging. Also for the device, the inhaler device specification should include the critical functional parameters. And I listed here a couple of these functional parameters to be included in the specification. Also, another recommendations. For the needle diameter, this device has a needle and a mesh, which used for inhalation of the dispersing of the powder out of capsules. We recommend the applicant to investigate the potential effect of the needle diameter and the mesh material on the product performance and powder residue in the capsule after testing APSD and DDU. For the button push in force, it's recommended to be determined over the lifetime of the inhaler containing the capsule in the chamber for consistency. And in some cases, some applicants like to also do the same testing without capsule to see the differences. The cap opening force, which is also critical, especially for patient handling. It's important to demonstrate if this force changes over the use of the unit, of the use of the device. And if there is if so, these changes, whatever it's reducing change or reduced force, is sufficient to keep the cap closed and open by the patient easily. My second challenging question: which of the followings are considered formulation development challenge in DPI? Fill weight, B, excipient grade, C, device resistance, D, amorphous content of sprayed APIs. The answer is B and D. Now, my third topic here is about the drug product characterization studies. Per the revised draft FDA guidance on MDIs and DPIs that has been published in 2018, there are a couple of characterization studies should be conducted on the DPIs as a one-time study to demonstrate the robustness and the performance of the product and to support the labeling instruction. Here in the right hand side, you can see I listed the characterization study that can be conducted, should be conducted for capsule-based DPIs. In general, DPI products units used for the characterization studies should be stored under conditions consistent with the storage conditions identified in the label. Also, to support the proposed shelf life at the intended storage condition, it's recommended that some characterization studies should be performed at the beginning and near to the end of the shelf life of the product's units, including in-use period, cleaning instructions, and robustness. Now I have some recommendation for a couple of characterization studies. Firstly, for the robustness, this is based on this specific product capsule-based DPIs. For robustness, it's recommended to assess the physical robustness of the capsules following the dropping and or vibration of the blister card for any damages. In the cleaning instruction study, it's also recommended to determine the drug residual at the end of the experiment for each unit, in addition to visual inspection for any capsule fragments and particle residuals. Fragment study. This is really critical for capsule-based DPIs, especially if the applicant is using material different from that of the RLD. So assess the puncturing and fragmentation behavior of the proposed capsule in comparison to the capsule of the RLD. Here is in this case, it's HPMC versus gelatin. Even if the applicant using gelatin, it's recommended to still perform this study to confirm there is no any fragments that can be inhaled by the patient. My last recommendation for effect of use study, where we are recommending the applicant to inspect the partially used product units after coming from clinical studies for any external damages. In addition, to evaluate the functionality and piercing mechanism of the clinical return inhalers for any mechanical defects or failures. Like, for example, the lack of rattling of the capsule, how to pierce the capsule, failure of pressing the button. This is really critical functional defects. My last challenging question here: which of the following drug product characterization studies should be conducted on the DPI product units at the end of near the end of shelf life? A, cleaning instruction; B, temperature cycling; C, robustness; D, in-use period. The answer: A, C, and D. I would like to end my talk with a general quality considerations for the primary DPI stability batches. Firstly, for the drug product constituent parts, the API, excipients, and device, it's highly recommended to use 3 different lots to manufacture the 3 primary stability batches. Drug substance discrete lots, the device constituent part components, as well as the critical excipients. And here, this is the carrier. For the batch size, given the complexity and the potential risk involved in scaling up the manufacturing process for DPI products, it's highly recommended to submit stability data from 3 exhibit batches at the proposed production scale. However, if this information is not available at the time of submission, it may be acceptable to provide at minimum 1 batch at the proposed scale and commercial scale, and to 1/3 of the commercial scale. However, if this is the case, it's highly recommended for the applicant to provide in-process and batch analysis data from several batches that's manufactured using the commercial proposed manufacturing process, like engineering, like developmental batches, performance qualification batches, to confirm the robustness of the proposed manufacturing process. At last, the product characterization studies should be conducted on the to-be-market configuration and version of DPI products, also 3 batches with reasonable sample unit to support the reliability, reproducibility of the product quality and performance. And here are the resources that can help with the DPI products: the FDA guidance, USP Chapter 5, and USP Chapter 601. At the end, my closing remarks here, again, DPI products can be more complex to review than MDIs and other complex products. So thorough characterize and understand the quality of the batches used in your exhibit batches, stability, and also BE studies and everything. Quickly identify the root cause for inconsistent DPI performance and optimize product design at early phase of the development to minimize the risk. Also, establish control strategy based on the evidence, scientific evidence gathered from thorough characterization of the stability batches and BE batches. At the end, I would like to acknowledge my office and my division, Division 5, my division director, Yi Ting, Deval Gaglani, my supervisor, and my colleagues in Unit 2. Also, I would like to acknowledge Office of Pharmaceutical Quality Research 2 in St. Louis, Jason, Rogers, Chining, and Nicholas. Office of Process and Manufacturing Assessment, Charmesta, Chining, Chu, and Vicky, and also OGD, Office of Research and Standard, Division of Therapeutic Performance 1, past and present members of nasal and inhalation product team. Thank you so much, and I'm looking forward for all questions. And I'm welcoming my other colleagues, my other speakers, and the panelists. Thank you.
All right, thank you again to our presenters for their fantastic presentations. We'll now begin our question and answer panel to include all presenters and additional panelists joining as well, which I'll introduce in a moment. But in the meantime, we want to go ahead and invite attendees in the room. Please line up at the mics if you have questions. We must have some questions after those really informative sessions, right? Yeah, okay. So as I mentioned, we'll have all our presenters here, and also joining the panel, we have Dr. Yili Lee, with Senior Pharmaceutical Quality Assessor with the Division of Product Quality Assessment 11, within the Office of Product Quality Assessment 2. And also Dr. Brian Newman, a lead pharmacologist within the Division of Therapeutic Performance 1 in the Office of Research Standards, within the Office of Generic Drugs. And also for those logged in online, if you haven't had a chance to submit your questions using the online or on-screen link and QR code, please do so now. And we'll answer as many questions as time allows. So let's go to our first in-room question here.
Thank you for the presentation. So for some of the peptide product that's approved way before the guidance is published, they may not necessarily have any unspecified impurities at 0.10% level as the specification. So would that play a factor in agencies' review when looking at the ANDA applications? Because if they can be allowed to be higher than that, but then we are holding to a much tighter spec and the golden 0.10 number.
So yeah, you're talking about if you're talking about peptides. So for those peptides covered by the peptide guidance, yeah, usually we ask you kind of like identify everything at or about 0.10% and identify control in the specifications. Also, there are some peptides not covered by the guidance, but however, if you look for PSG, the PSG will clearly state like this guidance applies to this product. We have similar expectation for the approval at the end for that type of product. Do you have any question for a particular product or just general questions for the?
Actually, if you don't mind, I'm going to jump in here, having a little bit of work, not as much as you, Lee, in the peptide arena. What we really look for peptides, while that's the guidance of 0.10, when it comes time to really doing the actual work and drug formulation, it's always the comparative analysis versus the RLD. So if you do your homework really well in P2, you're looking at the impurity profile of the RLD. And so if you see the RLD is having a higher limit than you would normally have according to the guidance, you can submit those 3 batches relative to the 3 batches that you have. And usually at expiration, you want to be doing that on the RLD, and you want to be doing that as far along as you can for yours as well. And you can provide a good justification, in which case the agency will consider that.
Great, thank you for those responses. We'll go ahead and move on to the next question in the room. Over on this side.
Hi, thank you for the presentations. I just want to note that the presentations that give challenges and potential solutions are extremely helpful to industry. And my question is related to some of the particle size data that goes into the applications. You know, we include all of these particle size testing data, and in module 2, where we do summaries, the agency has published some summary tables for doing particle size by MDRS, and I'm wondering if for particle size done by different methods, such as laser diffraction, are you also looking to have us generate some type of summary tables for module 2, or do you not care?
Okay, I answer in general. So in general, everything in the application has been shown and we should review everything.
Right.
So anything included in the P2 summary, yes, we look at it and review it. Any justification provided, anything we usually do. And if we have any concerns, we cite the deficiency to the applicant for more clarifications. That's in general. Did that answer your question?
I'm talking about the module 2, like 271. I'm just wondering if you want summary tables for particle size along with what's in the module 3.
Go ahead. Yeah, I think if it's helpful, definitely do that. I don't think it's a requirement, but if it's something that's going to help the justification, then you can do that.
And I may add here, I recommend everyone. So whenever you submit anything, you may think about, okay, it will be shown, or it's not important. Provide a reviewer guide. Reviewer guide is really amazing that you can put all the documents with hyperlinks and includes everything you need to. So we usually use this as a very guide for us, help us a lot to find whatever we need. And summaries and tables, that helps also a lot. That's in general.
Okay, thank you.
Again, we'll go on to our next in-room question on this side.
Yes, thank you. My name is Michael Lichminat. I just wanted to say thank you, FDA and CEBA, for having this training today. It's really informative and just gives the industry a better understanding of what the recommendations and expectations are. Just quickly, could be directly to Maria Flynn, Mrs. Maria Flynn, or just in broad for the FDA panel. I just wanted to ask, is there any agency updates to the IIG database, specifically to improve excipient toxicity level in pediatric patient within the oral suspension type, maybe oral suspension specifically dosage formulation, or in general?
I love the question. Thank you so much. So something I didn't have time to go over a lot of times with taste masking and a lot of the different formulations, like for suspensions, there's a lot of intricacies that can occur, and in each excipient can have different impurity profiles. And so he was talking about the IgG, which is the inactive ingredient database. And so there's a database, and you have to look at it not only for the actual exposure, but you have to think about it as the combined exposure of your different excipients. And specifically, in a lot of the excipients that are used, there's going to be trace amount of impurities, and we have a good sense of what those are. And it's really important to know your excipient impurity profile, because some of them are of a cohort of concern. They do have toxicological. So to your end, the database is as current as possible. So you do that, but when you do it from a holistic perspective, because there's going to be multiple excipients. And sometimes when you look at them, like for instance, flavoring, it's very important to get that information from whatever vendor that you're doing, like Tutti Frutti or something. For instance, there's a case today I'm thinking of in my mind where there was an issue with benzyl aldehyde, which is a cohort of concern. And it was in multiple excipients, not excipients, but ingredients of the flavor. And had they not looked at that profile, they would not have known that it exceeded what was normal and healthy and safe for use. So to your end, the database is as good as we have right now. And I believe they updated it. If you obviously, if there's something that's more helpful, and if you need to, please reach out to the agency so that we can include the information that you have or information that you're concerned with. And specifically, if you have something that you're trying to do and you're concerned about it, go ahead and submit those control correspondences to us. And if necessary, not in oral liquid situations usually, but in the more complicated situations, you want to use our pre-ANDA. Make sure you go through that pre-ANDA process if there's something really interesting and fun going on. And the sooner you communicate with us, the sooner we can help you and be proactive.
For sure, thank you.
Fantastic. We have another question here on this side of the room.
Okay, so the impurities above 0.1% should be ID'd for peptide, right? But to what extent the mass spec should, like the identification should be? Do you expect like full structure and sequence information, or can we just do a mass spec comparison and match to the ROD impurities?
Oh, yes. So that's a good question. We go back about the impurities. The general idea is, we encourage you identify everything at or about 0%, and then, do extensive comparison with the ROD. In that way, like we understand what's the differences between your ANDA and ROD. If you identify I think for certain impurities, only retention time, you say, oh, the same as RLD impurity, that's not sufficient. So we usually ask you to provide further characterization. For example, you say the same molecular weight or like MS fragmentation, the same as RLD. Then if you observe the RLD level is higher than your end product, then that means your limiter is qualified. However, if you observe the level in your product is higher than the RLD, then you may probably think about like immunogenicity mitigation.
Thank you.
All right, if we don't have questions in the room at the moment, we'll move to an online question, but I still want to encourage everyone here, don't be shy, still feel free to come up to a mic if a question pops in your head. So we'll go to our first online question.
All right, this is for Dr. Nashwa El-Gendi. How do you deal with polymorphisms in DPIs? Yes, that's correct.
DPIs in general. So for DPIs, this is a powder. So powder characteristic is very critical here. For the API and the carrier polymorphism for the API is very critical, and applicant should show if there is any polymorphism, they should have a testing for this polymorphism to be sure that the consistency polymorph used all over the drug substance lots. They have to provide all the chemical characterization or evaluation, PXRD, TGA or DSC, all the data for at least 3 lots of the drug substances with the polymorph that's consistent. Also, it's recommended to include polymorphic phase testing in the drug substance specification. For the drug product as well, the batches, it should be tested for any change in polymorphism throughout the shelf life of the drug product. This information we are looking for all the time.
Thank you for that response. Again, if anyone else has questions in the room, please come up. But in the meantime, we'll go ahead and go to our next online question.
All right, this question is directed towards Dr. Brian Newman. Recently, the FDA has published PSGs for DPIs that included other alternative in vitro studies that could be used for bioequivalence or product characterization. Can any of these studies be used to test the RLD or a proposed generic?
So that's a good question. I think, so what we can say now is that, of course, what's currently posted in our PSGs are current recommendations, but as was discussed yesterday during the presentations on alternative approaches, we're obviously been looking at both what study designs for alternative approaches will work for both MDIs and DPIs. So I would say that certainly some of these tests can apply for DPIs, and there was a good slide to show that across even with the MDIs, a lot of this depends on the characteristics of the product. So if you're considering this particular DPI, the API itself and its own characteristics, you can try to discern what types of alternative approaches might be applicable for that. So with that in mind, of course, you could then develop a alternative approach and provide that in a pre-NDA meeting request to get feedback from the agency. And certainly with considering this, I'd also point out that the PSG forecast list provides a good indication of how we plan on working through and considering the different PSGs, which ones needs to be revised for the alternative approaches. So if you're considering developing a product and you have this question in mind, I'd encourage you to take a look at the forecast list to see when we might be providing guidance and consider how you want to develop your program at that point.
Thank you. So this still looks like we're a bit shy in the room, so we'll still move on to our online question. Yep.
This question is directed towards the panel in general. If an API cost per milligram is very expensive for an ANDA applicant, does the FDA accept ANDA applications with smaller batch sizes other than the recommended ones? What is FDA's expectation?
So I'm talking from for example, for the complex drug product, we usually recommend 3 batches on the commercial scale. However, also again, we may accept 1 batch at a large scale and 2 batches, 1/3 of the commercial batch scale. And again, if there is any questions like this, we highly recommend the applicant to use all the FDA communication channels during the development stage. through the pre-ENDAs or CC with all the questions, and we are happy usually to help them with any with clarifying any question they have. Okay, thank you.
Thank you. So we'll go on to another online question.
All right, this question is for Tina Zhao. What are the recommendations for the samples used in comparative studies?
Yeah, for the test product, we ask for at least 3 batch of the test product manufactured from at least 2 lot of API tested on or near the at the release and at the end of the shelf life. And also we ask for at least 3 batch of the ROD, I mean, if available, at a different age before the expiry. And also in your submission, we recommend you provide a table to list the test product, the batch number, the API lot number, the manufacturing date, the expiration date, and testing date to help us to figure out the age of the sample. Yeah, that's it. Thank you.
Thank you for that response. Do we have another online question?
Yes, so this could be a follow-up for Tina Zhao and as well as to Dr. Yili Li. If I performed the comparability study aged and near release, it is recommended to update the study with samples aged at the end of the proposed shelf life. But which test needs to be updated? Only impurities profile comparison? Or do we need to also update the aggregate test? Or which tests need to be updated?
Yeah, I think all of them should be updated. Yeah, thank you.
Yeah, I just want to add, the characterization not only for impurities, but also for like high order structure and aggregations. So the study should be tested, should be performed near release and at the end of shelf life. We want to say like at the end of shelf life, your product behave like properly.
Yeah. Thank you again for those responses. And again, reminder to those in the room, this is your chance to come up and ask a question. All right, we've got someone on this side of the room.
I have a question for the panel. So you talk about accelerated testing. And in practical, there's a lot of different rules to use, maybe Arrhenius equation or the 10-degree double rate, those things. But we know that's not universal for everything. What thought accelerated testing you have to unify those things?
Can you repeat the question? Just
accelerated testing.
Oh, accelerated.
Yeah, accelerated testing for the shelf life. Like it's 2 years shelf life, you do 6 months at higher temperature. And what temperature you're going to choose, what time choose you based on some acceleration equations. Today we use some simple rule like Arrhenius equation or 10 degree for double rate, something like that. My question is not everything follow that equation exactly. And what's general thought to case by case to do that?
I can go on for this question. So I recommend you look at ICHQ1E. It states very clearly what situation, for example, you have, if you fail accelerate, what you're supposed to do is usually you need to start in intermediate stability. And also if intermediate stability increase significantly, what you're supposed to do. So there's a very clear decision tree in ICHQ1E. If you follow that, it will be clearly stated what you're supposed to do. And if the long-term stability is room temperature, what you're supposed to do. However, if it's long-term stability is refrigerated, what you're supposed to do. So I recommend you just go follow the decision tree in ICHQ1E. Do I answer your question?
Yeah, I agree with Ely. I feel like the guidance is very clear, but I want to bring up one special situation, and this doesn't happen. I have a lot of work done in the PEPFAR space, and sometimes in the PEPFAR space, we have zone 3 and 4. So when dealing with things that we know are going to different climates, such as very humid and, in the Southeast Asia or in the Caribbean or in Africa, in those situations, those are a few deviations that we would say away from the guidance. But in those situations, then they would have a higher, like 30 degrees and really high humidity, and sometimes. So those are the few exceptions, but for the most part, they do follow the guidance in terms of if there is an issue with the accelerated, then you would follow the guidance.
Thank you. And now we have a question on this side of the room.
Hi, it's me again. So you recommended the orthogonal method for the characterization, right, of peptides. So say if we have two methods, we try one of them is not working, like feasibility, it's not feasible. So should we just keep trying till we have a second method, or would that like one method be acceptable in that situation?
So yeah, usually we recommend like orthogonal method. So it's kind of like in cross-checking whether your results are like comparable to each other. Most of the time, so I don't see. Yeah, I think if two methods are not working, you should try more different type of method. And if you really exhausted all the technique and you only found one method working, I encourage you provide like a clear justification for everything, why, like state clearly why this method is not working, what's the problem with that. I would say it will be case by case, but in general, yeah, we do recommend that you have an orthogonal method.
So like say if like for aggregation test, right? Say if one method found no aggregation and then we do a paper-based like a risk justification, the other method, for example, SAC mouse is not working, do you think that can be approach we can take? Very specific, but
yeah, it's really like I cannot give you straight answer, like because it depends on the product, also depends on your result. As I said, if you think no aggregation detected with one method, you definitely need to provide a clear information, like whether your result is whether like your result is correct. So you definitely need to check. And I don't see, so for peptide, usually we recommend you do like high order structure also. Actually, if you have aggregates, sometimes NMR is can also be a good indicator, like whether there's aggregate. So NMR, high resolution, like two-dimensional NMR can like double check both the highest order structure and the aggregates, whether there's an indication. So I don't say like, I mean, so far I don't say like none of the method working for certain thing.
Thank you.
All right, we have one more question here.
Yes, I'd like to know, will the agency grant 36-month dating on an initial application, an ANDA, for a complex product if you have the 36-month data for both your product as well as the brand? And just to be clear, the size of the manufacturing that you've submitted is the size that commercial will be at. It's the commercial scale.
So your question is if the applicant provided 30-month stability data for his product and also the brand in the commercial scale, in the commercial manufacturing site.
Yes.
What the question in this case?
The question is, would you grant 36-month expiration dating?
It depends on the data. If the applicant propose 30 months shelf life, we will work on that to see if it's acceptable or not based on the data provided. If the applicant provided 24 months and they have 36 months, but they propose 24 months, we'll not tell them, yeah, you have until 36, you can extend it now. No, it's the applicant decision. But the data provided based on what the applicant provided, what he is proposing. You having your application in P8, you're proposing 24 months shelf life. This is our shelf life. Extra data, we review it for sure, and if it's adequate, but we accept 24 months based on the proposal provided to us.
Right. Okay. So you can, if you have 36-month commercial scale, it seems the agency would take that into consideration. And
it's reviewed. Everything is reviewed, but at the end, the product approved based on the proposed by the applicant. Applicant proposed 24 months. That's the shelf life. Yes, if it's everything is fine, it's fine. If applicant would like post-approval, they can provide a request to extend the shelf life or whatever based on the data already provided. Or if they feel that the product already up to 36 months adequate data within the specifications, they can request to extend the shelf life to 36 months.
Okay,
it's up to the applicant. Okay.
All right, thank you so much. Looks like that's all the time we have questions for, or all the questions we have time for. Thank you again to our presenters and panelists for all the excellent information and insights. We'll now be taking a break which will resume at 11 a.m. Eastern Time. So thank you again. Let's give them a hand. All right, we're ready to get started again. Everyone's coming back to their seats. So welcome back from the break. We hope everyone was able to take some time to refresh and came back ready for our next set of exciting sessions. It's now my pleasure to introduce our speakers for the next sessions, continuing the spotlight on generic drug review challenges and solutions. So we'll be hearing presentations from Dr. Yang Liu, a senior staff fellow within the Division of Bioequivalence 3, Office of Bioequivalence in the Office of Generic Drugs. We'll also be hearing from Dr. Feng Wu, a senior pharmacologist in the Division of Quantitative Methods and Modeling within the Office of Research and Standards in the Office of Generic Drugs. We'll also hear from Dr. Liken Liang, a supervisory chemist within the Division of Product Quality Assessment 10 in the Office of Product Quality Assessment 2 within the Office of Pharmaceutical Quality. And we'll also hear a presentation from Dr. Yang Wang, lead pharmacologist, acting deputy division director within the Division of Therapeutic Performance 1 in the Office of Research and Standards in the Office of Generic Drugs. So please join me in welcoming our next presenter, Dr. Yang Liu.
Thank you for the nice introduction, and good morning, everyone. It is my great pleasure to be here again, sharing some case study experience with all of you, both in person or online. So the story we want to share today, together with my dear colleague, Dr. Fang Wu, is the totality of evidence, including a PBPK modeling to support the BE assessment and the approval of mesalamine DR tablets. So hopefully through today's presentation from both of us, we can deliver the following messages. Number one, we want to present this case study of an ANDA for mesalamine DR tablets with some dissolution issue in some certain dissolution media. And also we try to share with you and help you understand OGD's BE evaluation based on the totality of evidence for this case study. And then Fang will share some detail about the PBPK modeling and how we are using it as a critical evidence to support the final approval of this ANDA. So here's my disclaimer. This presentation reflects the views of the presenter only and should not be construed or represent FDA's view or policies. So first of all, let me just give a very quick introduction or a background refresh about the ROD product and what are the studies recommended by the PSG. So the ROD product is Takeda's Lealda DR tablets, 1.2 gram. It was approved in 2007 under the NDA number 22000. This product is indicated to treat colitis in both adults and pediatric patients. And here's the PSG. I just took a quick screenshot of the study recommended by the PSG, which is published in June 2016. There are 3 pivotal studies recommended for developing a generic version of this drug. An in vivo BE study under fasting and under fat conditions respectively. And the third study is an in vitro dissolution study. And I do want to highlight one thing is the design of the in vitro dissolution study is it actually includes 3 stages. The stage 1 and the stage 2 are called a pre-treatment or prepared stages. And the evaluation stages are recommending using 4 different medias at pH 6.5, 6.8, 7.2, and 7.5 respectively. And in a later slide, I'm going to discuss why this is designed as such and what is the physiological reason. And now let's just dive into, because due to the time, we just dive directly into the case study. So in this ANDA, based on the data submitted, the reviewers found that the PK and statistical results for both the pivotal in vivo studies met the BE acceptance criteria. I do want to say one more thing about the in vivo study recommendation is not only the Cmax and AUC overall were evaluated, the PSG also recommend to evaluate the comparison, the comparison between the test and the reference drug for the AUC 8 to 48 hour, which is the partial AUC kind of representing the drug absorption in colon, where is the site of action. So that also brings up the question about the mechanism of this drug or the site of action. So this drug, mesalamine DR tablets, is actually a local acting drug. Is used to treat ulceritis in the colon. However, the function is not done through like the plasma concentration. Is actually dependent on the local concentration of mesalamine delivered into the colon directly. So in this case study, the test formulation, it's going so fast, but anyways. The test formulation adopts similar mechanism of drug release. Is actually using the same pH-dependent release controlling excipient as the RLD. However, the dissolution profile, not all of them, but some of that actually are very, very different between the test and the reference product. I don't know, it's keep flashing. But we can see actually over here, I don't know. So, but this is just a quick review. The left two figures, the profiles, are actually the comparison for the lower pH values, 6.5 and 6.8. And the last one, which is very comparable, is for the 7.2 and 7.5. And in this table, we can also see the calculation for F2, or the dissolution similarity. We can clearly see that the dissolution are not that similar in the low pH conditions. And we're wondering why, and how big of an impact it actually caused in our evaluation for bioequivalence. So let's just go through why this actually is, the drug itself is, why is keep going? I didn't touch anything, but anyway. Okay, so this is the mechanism for this coating, because if the drug is not coated due to the quick dissolution and quick absorption of mesalamine, the majority of mesalamine will be absorbed before it actually reach the site of action, which is a colon. And on the right side, we can actually, I'm bringing the GI tract into three sections in terms of the fate of this drug. In the stomach and the duodenum, where the pH is relatively low, the coating will protect the mesalamine from early release. And in the middle section, which is the small intestine, the outer coating will be eroded and start to release the drug slowly, and it will still deliver most of the mesalamine APIs into the column to do its function. So in this slide actually is showing, the coating for this product. It's showing the function. It's actually telling the same story. Let's go back here. So it's the same thing. So the pH dependent or pH protection of the outer coating will make sure the drug stay intact in the stomach, and it will start to erode, as you can see on the left side. It start to erode in the duodenum, and mostly were eroded and start to release the drug in small intestine. And per the RLD label, it also says the coating of the pH-dependent polymer film, which breaks down at or above pH 6.8, normally in the terminal ileum or terminal small intestine, and start to release the mesalamine. And how does it serve the function to make sure the drug mesalamine is actually released slowly throughout the whole large intestine or whole column? It's actually based on another mechanism. But before that, here is another piece of evidence from the radioactive study showing where the drugs are actually released. As we can see in these figures, in the first figure is the T3, 3 hour, which is 3 hours after administration. We can see at the distal small intestine, we can already start to see some fuzziness around the tablet, which means the mesalamine, the mesalamine was labeled, the mesalamine drug started to release from the tablet, which means the outer coating is pretty much eroded and does not do keep the tablet intact anymore. And as the drug goes into the small intestine, the large intestine, we can see the drug already start to go everywhere and start to do its function. And that's exactly what we are actually concerning about, is whether it is similar between the in vitro dissolution phases and the physiological conditions, whether it is representative. As we can see in the the green bars. And we can see the drug actually go through the physiological conditions. It will be eroded, the outer coating, the red outer coating will be eroded after it exits the small intestine. And when it goes into the distal column or into the column, the outer coating is pretty much gone. However, that is a little bit inconsistent with what we actually designed the in vitro study. Again, the in vitro study design at different pH levels is try to represent all the conditions that we may appear or the drug may experience in the GI tract as it delivers. And the difference here is for example, for the pH 6.8 condition, which is used as the evaluation stage, the drug actually didn't have a chance to be exposed to a higher pH value condition, just like in the small intestine in vivo. So our question is whether this is actually making a significant physiological impact. So the question is to correlate the physiological and PK and dissolution study as one big piece of evidence. What kind of tool can we use? And Fang will be discussing about that. But now I just want to give a very quick summary of what we have observed before we actually having the PK modeling kicking in. So what we have observed is the in vitro dissolution issue for pH lower than 6.9 conditions, and we are starting to see different dissolution profile between the test and the RLD product. And the in vivo drug dissolution released through the GI tract may vary due to the physiological conditions in different patients or healthy volunteers. And we actually recommend the applicant to conduct the the PK/PD modeling, IVIVC correlation, and to use that as a supportive evidence to justify the lower pH value incomparable dissolution result. And that's where I'm going to stop and turn that into Fang. But before I do that, here is a quick question, is based on the data we have collected, either directly through the RLD labeling or through other literatures and evidence, is that true that the RLD product, the Leota DR tablet, will stay intact and prevent the drug release until it actually goes into column? Apparently, the answer is false. So this is not the case, especially due to the radioactive study. The drug already starts to release in small intestine. And here is, I just want to steal like a couple of seconds from Fang. So here's my acknowledgement. I first want to thank Dr. Partha Roy, the OB director, who is very supportive about, this is a very interesting case, and he's, the leader for the office level discussion. I also want to thank DB3 management, April, Key, and Li Gong, who actually having a lot of great discussion with the assessment team and make final decisions. And OB management also provide very helpful insights and questions and comments through the whole story that I'm sharing today. Last but not least, we have the great supporting team from the DQMM, and I will let Fang to introduce more about the PK/PD modeling part. Thank you very much.
Thank you, Yang. Next, I would like to continue the second part of this presentation. I will introduce how the PBPK modeling can serve as a part of the totality of the evidence to support approval of the mesalamine DR tablets. Here is my disclaimer. And learning objective, for my part, I will mainly explain how to develop PBPK model to predict a local drug amount by incorporating dissolution data at different pH. First, we want also to identify which dissolution is the biopredictive. A little bit of background on the GI locally acting products. As we know, assessing the BE for GI locally acting products is challenging because the systemic exposure may not reflect the drug concentration at the site of action. So the BE recommendations of oral locally acting drug products are based on the drug product properties and the mechanism of action. PBPK models may be used to explore the correlation between dissolution in GI tract and the plasma concentration profiles and predict the local drug amount and support BE evaluation of such products. As Yang mentioned about these background of the mesalamine DR tablets, it is indicated for colitis. And the product-specific guidance for this product recommends fasting and fat PKBE study, and also the comparative dissolution studies at different pH, of course, include a pretreatment stage 1 at pH 1.2 and pretreatment stage 2 at pH 6.4, and then evaluate the comparability at each different pH, 6.5, 6.8, 7.2, and 7.5. However, for this generic drug product, we found the F2 values less than 50 when comparing the dissolution between test and reference at pH 6.8 and 6.9. However, it's more than 50. It's comparable when the dissolution is over pH 7. But the test product and ROD were found to be bioequivalent for systemic PK under both fasting and fat conditions. Here comes the question, whether the slower release of test product at a certain pH would significantly impact the local drug exposure compared to ROD. And you have seen these PSG recommendation and know the conditions we recommended for conducting the in vitro comparative dissolution study. And in this, for this generic drug product, we found that at pH 6.5, 6.8, 6.9, the dissolution are not similar between test and reference, and the F2 is less than 50. You know that we use these in vitro dissolution testing over a range of pH to serve as a surrogate of in vivo drug release in the GI tract. And we see actually these test product shows decreased release compared to ROD at pH less than 7, but it's comparable at pH over 7. Whether these will cause any significant impact on the BE for this local drug, GI locally acting products, we will use PBPK modeling to support providing some evidence. And we use the PBPK modeling to try to identify which dissolution condition is biopredictive. And we will also use PBPK modeling to predict the local drug amount in the colon and use population simulation to predict the percentage of drug absorbed in the colon. So the so here I would like to use a little bit of this flow chart to go over the how we develop, validate, and apply this PBPK model. We first use the intravenous PK data obtained from the literature to estimate disposition parameters. Then we develop the PBPK model for mesalamine DR tablet. The intestine first pass effect was optimized. And then we also incorporate 3-stage dissolution data with pH 6.8, 6.97, and 7.2 as stage 3. Incorporate them into PBPK model and predict the PK, systemic PK. And then the PBPK model was validated using dissolution and the PK data obtained from literature or other ANDAs, and to validate this model. And then after validation, we use this PBPK model to see which one is which dissolution is biopredictive and predict the drug amount in the colon. These two figures shows that PBPK model can capture the PK well after IV bolus administration of 250 mg mesalamine. And it can capture the PK after oral administration of the delayed release tablet. And we use other data from literature, and we can also see the modeling predict well of the PK. And we, as shown on the right table, we use the data, dissolution data, and the PK data obtained from the other ANDAs, and we show that the PK prediction actually is pretty well, and with the prediction error less than 20%. This means that the model is validated using these additional data. Then we use the validated data to evaluate whether the dissolution data at a certain pH is biopredictive. As shown in these tables, we can see if we use the dissolution at pH 7.2 and 7.0 at stage 3 incorporated into the PBPK model, then the prediction error is less than 22%, which means these conditions are biopredictive. But if we use the dissolution at pH 6.8 at stage 3 into the model, then we cannot predict PK well with the prediction error more than 30%. So we know that the dissolution at pH 7 and 7.2 at stage 3 are biopredictive. So then we incorporate this biopredicted dissolution into the model and predict the local drug amount in the colon and also the percentage drug absorbed in the colon. As you can see from the right top table, and the simulation suggests that the local amount in colon is similar between R and the test product, ROD and the test product. And the population simulations showed that percentage of drug absorbed in the colon is similar between the ROD and the test product, with 90% confidence interval of this TR ratio for the drug absorbed in the percentage of drug absorbed in the colon can fall within 80 to 125. This should demonstrate there is a low risk of non-BE at this site of action for the generic drug product. These two figures you have seen previously with Dr. Yang Lu's presentation, and based on the formulation design, release mechanism, and GI physiology, shown here, we know actually the drug, the generic, both RD and the generic drug, they have the enteric coating. The coating will be break down after it enters in the small intestine, especially the terminal ileum, where the pH is 7. Then the core continues to pass into the GI tract and go to the colon and continues to release the drug. The colon, you know that the pH is around 6 to 6.7. And we can also see the drug, actually, in the scintigraphic figure, the drug release in the ascending column is at the ileum, terminal ileum. It's kind of consistent with what we are thinking about the release mechanism. These are also consistent with our PBPK modeling. And the PBPK modeling shows that the dissolution at pH over 7 is biopredictive, but not for the dissolution at pH 6.8, because the drug is already break down before it enters into the colon. So the pH 7 is more biopredictive, as demonstrated by PBPK modeling and also the release mechanism. So in summary, the PBPK modeling simulations supported that the 3-stage dissolution profiles at both pH 7, 7.2 at stage 3 may be biopredictive to the local and systemic exposure. The predicted amount of mechanism in colon and the percentage of drug absorbed in the colon, they are similar between ROD and the test product. So the totality of evidence based on the dissolution data, formulation differences, gut physiology considerations, along with the PBPK modeling results were used to conclude that this generic drug product has low risk of bioinequivalence at site of action compared to the ROD. So this work, as mentioned by Dr. Yang Lu, supports the approval of this mesalamine generic drug product. So here comes a challenge question. To evaluate the BE for locally acting products, the evidence could include the following: A, formulation and the dissolution comparison. B, gut physiology considerations and release mechanism. C, PBPK modeling and simulations. D, all of the above. I will let you think about 1 minute. Yeah, 1 second. Then the answer is all of that, considering the time limitation. With that, I would like to acknowledge our colleagues in DQMM and also the reviewers from Office of Bioequivalence, and especially Dr. Sharon Thomas, who did most of the modeling work, and the reviewers from OB, Dr. Yang Lu, Dr. Teresa Lee, and Gong, and they are valuable inputs and collaboration during the review of this generic drug product. Thank you. Then I would turn over to Dr. Li Kan Liang. Thank you so much.
Okay, good morning, good afternoon, and good evening. I'm Li Kan Liang. So today I'd like to discuss the complexity of generic oligonucleotides, some key challenges in achieving and demonstrating sameness, and also impurity profiles and controls. And finally, I'd like to share with you some of the agency's efforts in addressing these challenges. Here's my disclaimer. The presentation reflects the views of the presenter and should not be construed to represent the FDA's views or policies. Oligonucleotides are short strings of nucleic acid with therapeutic effects. They can be fully synthetic or semi-synthetic, and they can be modified or unmodified. There are many ways to modify oligonucleotides, and modification could be at the 2' position or the 3' position of the ribose ring, or could be on the bases. And the oligonucleotide can be conjugated at the 5' end and the 2' position or the 3' end. The internucleotide linkage can also be modified, and in most cases, this modification is non-stereo specific, therefore leading to 2 to the nth power of diastereomers. This significantly increased the complexity of oligonucleotides. The ribose moiety can also be replaced with a morpholino ring. And the non-stereo specific modification by the PDA or the phosphorodiamidate in those linkages also leads to 2 to the nth power of diastereomers. I apologize for the busy slide here. This table summarizes the 20 oligonucleotides CDER has approved as of July 2024. The last column lists some of the complexity considerations based on the for generic oligonucleotides based on the RD labeling information. The PS and PDA modification of the linkages actually gives rise to from a few diastereomers to more than 1 billion diastereomers. The same regulatory requirements for generic small molecule drugs also apply to generic oligonucleotides, such as having the same clinical effect or safety profiles and identical active drug ingredients, which translates into active ingredient sameness for complex APIs such as oligonucleotides. Sequence chemical structures, diastereomeric composition, high order structures, and potency and activities, etc., are among the sameness considerations. For most synthetic oligonucleotides, sequences are well defined and controlled and can be verified. Regarding the structures, for the correctness and completeness of the modifications, including the position of the modifications, need to be demonstrated for generic oligonucleotides. The sameness evaluation also includes physical chemical characterization at the drug substance and drug product levels. Oligonucleotide impurities have some unique safety considerations, such as off-target effects, immunogenicity and inflammatory risks, etc. These risks need to be considered in impurity profile comparison and also impurity controls. It is recognized that binding of oligonucleotides to their targets are largely sequence specific. Then why is diastereomeric composition sameness required for generic oligonucleotides? This is because clinical evidence is not available to confidently negate the impact. And there are also some evidences indicating that stereochemistry and conformation of the oligonucleotides may impact their binding affinity and resistance to nucleus. For example, Iwamoto and others studied myeloimmersion and various stereochemically pure isomers and found that stereo configurations at the PS linkages substantially impact the pharmacological property of the oligonucleotides in their studies. Also, approved oligonucleotides counted all diastereomers as the part of the active ingredient. Therefore, unless adequate data is available to prove that it has no impact, diastereomeric composition sameness should be demonstrated for generic oligonucleotides. The graphs in this slide were adapted with permission from the ACS publication by Russell and others. The authors changed the RNS ratio at some selective internucleotide linkages of the 20-mer by using different activators. Several different methods were used to analyze the samples. Each curve on this slide represents more than 500,000 diastereomers. The first method did not give much information about the composition. Other methods gave, with wider peaks, with some distribution features. Let's look at the MICC method and compare the levels of profile changes from sample 1 to 2, and from sample 2 to 3. The levels of profile change look similar. However, from sample 1 to 2, the RS ratio was changed at only one additional chiral center. But if you look at samples from 2 to 3, the RS ratio was changed at 4 additional chiral centers. This comparison indicates that certain diastereomeric composition changes may not be proportionally reflected in the profile changes by some methods. To address the analytical method limitations, one may consider using multiple analytical methods with orthogonal separation principles that can provide detailed distribution profiles, combined with other orthogonal methods such as an NMR and with additional physicochemical characterizations such as the melting temperature or TM of an appropriate duplex from the oligonucleotide. The methods should be adequately validated for sensitivity, specificity, and resolving power. If the diastereomer number is low, one may develop methods to separate each diastereomer from other diastereomers and impurities. This way, one can compare the composition directly. To demonstrate sufficient sensitivity and specificity of the methods towards various diastereomeric composition changes, one may consider making multiple suitability test standards for method development and validation. These standards may have RNS ratios changed at a diverse set of a small number of chiral centers. If the methods are sensitive to such diverse and verifiable small changes in diastereomeric compositions, it is more likely that these methods may be able to detect the diastereomeric composition differences between the test product and the RLD. So for distribution profile comparison, one may consider comparing both overall and individual regions of the profiles. The acceptance criteria for sameness comparisons needs to be justified, ensuring comparable safety and efficacy profile as the RLD. The RNS ratio measurement at each of the chiral center after its creation is actually an indirect but effective approach to obtain stereochemistry information of your product, especially when the number of diastereomers is very high. And typical analytical methods may not be able to provide a precise picture of the diastereomeric composition. So it is important for firms to understand the baseline of their product for future potential changes and to ensure manufacturing consistency. This may be applicable to both the new and generic oligonucleotides. Typically, oligonucleotide impurities profiles are very complex with high number of impurities. Some impurities may be due to the incomplete modification or having extra nucleotides or missing nucleotides somewhere in the sequence. The complexity is further compounded by the high number of diastereomers in many cases. The large molecular weight of impurities makes it challenging to separate impurities with small structural differences. The resolving power of a single analytical method may be very limited, and co-erosion of impurities and isomers needs to be addressed. Oligonucleotides are excluded in ICH guidelines for impurities. The FDA has not published a guidance on impurity thresholds for oligonucleotides. And furthermore, the agency has not recommended structure-based classification of oligonucleotide impurities for safety evaluation and quality control purpose for generics. For impurity profile comparison and controls, multiple methods with orthogonal separation principles may be needed to address the co-erosion issues. The nature and location of the structural change in the impurities need to be determined. For example, for inotertion shown in this slide, an impurity with one methyl C unit missing could have 3 different sequences or structures. The exact location of the missing unit may need to be determined unless otherwise justified. If there's any ambiguity, one may want to see if it helps by conducting a comprehensive risk assessment and consider all the possible structures or impurities based on your process understanding. For impurity level controls, RLD comparison is the rule of thumb for generic oligonucleotides. Orthogonal and complementary analytical methods may be used. And when dealing with new impurities, higher impurity levels, proposed impurity thresholds, or impurity grouping approaches, comprehensive risk assessment may be needed to evaluate their impact on safety and efficacy. Off-target effect, immunogenicity, and inflammatory risks, and general toxicity, and any other risks associated with the possible impurities may need to be considered. Summation-based grouping acceptance criteria without comparing and controlling the individual impurities levels may be hard to justify for generic oligonucleotides. Some examples including the retention time segment-based grouping, or N-1 or N+1 groupings, etc. Also, the fact that oligonucleotide impurities have higher molecular weights and therefore lower molar amounts than in the small molecule drug case may not be an adequate justification for requesting a higher weight percentage limit in the oligo cases. You may want to see if a comprehensive risk assessment based on the worst case scenarios may help justify some of the grouping approaches. For RD comparative studies, 3 batches are needed at the drug substance and drug product levels. For evaluating the API from the RD products, one may obtain the API by an appropriate sample processing method, such as extraction or dialysis, etc., depending on the need and the nature of the RD comparative study. For diastereomeric composition sameness evaluation, sample processing should not change the diastereomeric composition of the sample itself. If a test is sensitive to formulation or process history, one may consider subjecting both the generic and the RD samples to the same process. For intrinsic property characterization, formulation impact may need to be eliminated. For example, TM is an intrinsic property of a duplex, but can be impacted by the presence of ions, etc. Therefore, TM measurement may be done at the drug substance level. For siRNAs, TM measurement may be performed at both the drug substance and drug product level to understand the formulation impact and also demonstrate sameness. Sameness evaluation on 3 exhibit batches at release and shelf life, at end of shelf life may be necessary. To support initial ANDA filing in a timely manner, one may consider evaluating a development batch for age considerations and submit the results as additional supporting data, provided that the development batch is equivalent to the exhibit batch in all aspects of CMC. CDER has various programs to facilitate and advance generic drug development, assessment, and approval. Some of the examples are listed here. The agency has published quite a few product-specific guidances on oligonucleotides to help generic product development. The agency also has internal and external research programs to advance the regulatory sciences. Here's an example of the research by our colleagues at the Office of Pharmaceutical Quality Research to address the challenges in analyzing co-eluting isobaric impurities. These impurities were difficult to differentiate and quantify by the conventional methods such as LC-MS. The OPQR work actually illustrated illustrated the possibility of quantifying these very challenging isobaric impurities. With that, I'd like to thank all the people and teams and officers listed on this slide. And here are some questions for you to consider. Question one, ON impurities have much higher molecular weight than impurities of small molecule drugs. And the molar amount of the ON impurities are lower, and therefore ON impurities should generally be given higher weight percentage limit than impurities of small molecules. True or false? So the answer is false. The next question, which of the following statement is true? Oligonucleotide binding is sequence specific. Stereochemistry of the OM modification need not to be considered in sameness evaluation. B, I can use 3 development batches of the generic OM for sameness evaluation and characterization for my submission, even if they are not true representation of the exhibit batches. And C, for complex sameness or comparative impurity profile evaluation, I can use multiple orthogonal analytical methods that gives quite different and detailed distribution curves or profiles on the same sample. Or D, currently I can justify my quality control of impurities in generic OM based on literature information about OM impurity classification without comparing them to the impurity of the ROD. So which one is correct? C, that's right. So to summarize, ROD comparison is the rule of thumb for generic ONs, and when necessary, use multiple orthogonal methods. Also, conduct comprehensive risk assessment to help justify proposed solutions. And as always, seek agencies' help via various channels. And thank you for your attention. Now I hand to Dr. Wang.
Good morning, everyone. So in the next 20 minutes, I will try to walk you through the journey of first approvals of several complex generic long-acting injectable products. Hopefully, I will tell a good story. So at the end, you'll be able to describe different types of long-acting injectable products to recognize what are the regulatory and scientific challenges we have experienced, and some of them have been resolved, and we certainly recognize there are remaining hurdles, and have better understanding on what is the product-specific guidance and their role for supporting generic development. And lastly, but not the least, to be able to have a better sense of how the importance of GDUFA-funded research program and how much that really contributed to where we are today. So long-acting injectable products, those are the products that are designed to provide controlled and sustained release for days and months. So in this slide and next couple of slides, I'm sharing information data that published in a review paper I co-authored with my colleagues, Dr. Wenlei Zhang, sitting in the audience, and 2 industrial peers back in 2021. We aimed at giving a very comprehensive overview of the landscape of long-acting injectables, including new drugs and generics in 2021. So the figure I'm showing here to show you the LAIs and the therapeutic areas that have been used. And you can tell they are very, very beneficial in the therapeutic areas that would require chronic disease treatment or management, because they can significantly improve patient compliance. Compared to conventional dosage forms such as tablets or capsules, we have far less numbers of applications approvals, but then the complexity level and the diversity of dosage forms is also very impressive. So as shown here, I'm gonna walk you through a little bit like history of the LAIs here. So back prior to 882, the very first generation or generations of LAIs could trace back as oil solutions and then some micronized drug substance suspensions such as penicillin G. So back then, it was already probably very challenging to bring those drug substance into a product that can be given to the patient. But I don't think back then, now it's 40 plus years ago, the scientists then had really clear concept of being long-acting drugs. I think the very first, by definition, long-acting with a very defined therapeutic dosing regimen should be the polylactide co-glycolide PLGA-based microsphere product, Lupron Depot, which was approved in 1989. So followed on that in terms of dosage form, in 2009, there was very first multivesicular liposome product. But that one was to deliver cytarabine, and then after that there was another MVL. But those two products, unfortunately, were very short-lived, not due to safety or efficacy, maybe more due to business reason. Then in 2009, going back to PLG-based delivery system, the very first in situ forming gel or implant was brought to the market as Eligard to deliver the same peptide leuprolide acetate as the microsphere product in 2009, as shown on the top right panel. And that product is organic PLGA polymer solution, and the peptide is suspended in the organic given as an injectable suspension. Then upon administration, the dosage form would form a depot on the site. Then after that approval in 2011, the history always repeats itself, right? So it goes back to the drug substance suspension. And this time it was for paliperidone palmitate, which is antipsychotic, and it's a nano size instead of micro-sized suspension product. Follow that, MVL came back as what was mentioned in the commissioner's opening remark to market. This time is a very, very successful product for pain management, the bupivacaine multivesicular liposome. So in the 2021 paper, when we worked on it, we had a table to list all the high revenue LAI products. And back then, we still did not have any generic products approved. But that time we were already hopeful because after that time was almost close to 10 years work, we already have in-house ANDAs that were under assessment. So we were very hopeful to see the first approval, which actually not one, we had three. We had three first generic approvals for LAI products for three separate ILDs. So those products were Sandostatin LAI, which is a peptide-containing microsphere product, and risperidone Costa, containing risperidone for antipsychotic treatment, and Vivitrol, which is an naltrexone microsphere product. So the Sandostatin LAI product is the product that containing a glucose PLGA polymer, which is very unique. Majority of the microsphere products on the market are using linear PLGA as shown here. So in the next couple of slides, I'm gonna share some of the regulatory challenges and scientific challenges we have had. So the regulatory challenge for PLGA-based LAI generics, because those are injectable products, parenteral. So per regulation, they need to be qualitatively Q1 and quantitatively Q2, the same as the reference listed drugs. However, for complex polymeric excipients in general, if you work with polymer, you know it's never a clean cut, right? The characteristic of the polymer is often a distribution, for instance, for the molecular weight. And the composition can also have certain variability. But then when it comes to PLGA, another layer of complexity is the products that are made by PLGA are really products by manufacturing. So depending on the manufacturing condition, methods, the raw material you start with may not represent the polymer you have in your finished product. And I'm having this figure here. It's in, I think, the Lupron Depot patent by Alchemist, showing you, depending on also your commercial scale, the level of degradation PLG can undergo is very different. So the regulation tells us, okay, you need to have the same inactive in the generic product, but it doesn't define what is considered as Q1 the same, and it's up to us to use the science to inform the decision making. And when we first started our research back in 2013, with the support of implementation of GDUFA, we knew nothing. That was the truth. So that regulatory challenge is translated into our scientific challenge. One, how do we define polymer sameness? What to do? PLG was not new, and PLG was very well known for formulation scientists. It started as a medical device for suture, and pioneered by, I think, Dr. the Lambert in MIT to be able to make high molecular PLG that is suitable for drug delivery. But most of the time, people do it to develop formulations by error and trial and error. So there is really no very systematically methods available to characterize the PLG and to inform. So we took the challenge and we funded our research program to look how we can reverse engineer the complex drug products and to identify the PLG and determine its characteristics. And we publish our results in a timely manner to facilitate generic development. Then I mentioned one of the first 3 approvals, the octreotide sandostatin LAR, used a glucose core branched PLG polymer. And there was 0 literature published back then discussing how we can potentially characterize this particular polymer and to prove if the glucose is really in the core. And when we first started working on this project with Dr. Ian Park at Purdue, they just used the conventional NMR, and we didn't see any signal. So we were like, what's going on? And is it really a glucose core branched polymer? And how do we characterize the branch frequency? All those things were very challenging. And honestly, when we first started, we didn't know if we'd be able to deliver, but we have to start and just keep going. But at the end, we were able to actually publish multiple papers providing characterization methods to characterize the branch structure of the polymer. And we also actually characterized all the commercially available raw materials to guide the generic development. Then, so this slide just summarize what we have learned and our current standard to guide the Q1 polymer sameness assessment. So our expectation is we would like to see the polymers extracted from the finished products, both T and R, to compare their properties to ensure they have comparable characteristics. This is not a conventional approach or a routine ask for all polymeric excipients. This is mostly, like I mentioned, we want the applicant to really keep the endpoint at the starting line to realize your game is not simple. Sometimes we see data submitted on raw material, but if you are really having a raw material matching on face value, the properties in the finished ILD product, when your material goes through manufacturing, you may not end up with a product that has comparable performance. So it's really for the applicant's benefit to know this in advance. Then the Q1 sameness, at least for most of our products, we think we have done great progress and have resolved most of the difficulties, if not all. But then in recent years, we started seeing, because PLG polymers, they have well-established clinical safety profile. So whenever a company can leverage from this group of polymers, they tend to stay within this group of polymer without trying new excipients. But then it's not sometimes you use one kind of polymer could give you the desired release characteristics. So then there are plenty of smart people, right? So what they do is they start to mix different PLJ polymers to fine-tune the degradation profiles to get what they want in terms of the release characteristics, which is brilliant. But then it's also going to be tremendously challenging for generic perspective. The first question we have is, if there's a mixture of two PLJ polymers, and how is it scientifically feasible for us to actually separate them? And if we can, to what extent? And what would be the most reasonable regulatory expectations in terms of the sameness in generics? So sometimes we do research, and that could, on face value, scare generic industry in terms of what are you trying to do. But sometimes we do things to the bottom because we want to know where should we draw the line. So we do something doesn't mean we're going to require the exact same thing as part of the generic development. But sometimes it's actually the opposite. If we have enough understanding, for instance, to show you don't really need to have the very detailed level of separation, and as long as you can meet the bulk property, that's sufficient, then that's our internal justification to develop the policy. So this is still ongoing, and we have made good progress, but we are not there yet in terms of clearly articulate where we stand when a mixture is used in a formulation. So next, the scientific challenge is the in vitro release testing. So this is a general challenge for long-acting products because the IVRT itself just very long, and because the release mechanisms also can be very complicated. So sometimes depending on the method condition, for the same formulation, you can get very, very different profiles. I think yesterday there was a good discussion in the IVRT session, right? Talking about then what should we aim for? Should we aim for like a profile condition that mimic the in vivo PK? Or should we have a condition that we know that is more discriminatory for differences in formulation characteristics, composition, or manufacturing parameters? I think those are all very good and valid questions. And that's part of our research to really first to explore all the scientifically sound methods, what are out there, and then what may be more suitable for QC versus for BE purpose. So here for long-acting injectable, we have been working on, for instance, looking into real time and then also accelerated time because we understand how time is valuable for drug development. So we looked into develop important tools to understand the impact, how the formulation parameters like the source of the polymer, if you purchase from supplier A versus B, if they give you the same performance, and then also use that to explore novel B approach. For PLJ-based microsphere products, we are not there yet, but we have ongoing research for instance, the in situ forming, which we are trying to see if we have enough understanding to push for totality of evidence-based in vitro only approach for supporting BE, or as a in between, if we can have in vitro in combination with in vivo, then reduce some level of burden at the clinical site. And ideally, once we have methods, IVRT methods that we're really comfortable, we will start to maybe implement them into our PSG to be more descriptive so company can follow. And we also start to include references in our PSG so company can directly look at the papers we have published. So challenge 3, bioequivalence study, is already sort of part of the IVRT. In general, the BE study, our expectation it should be accurate, sensitive, and reproducible approach. So for PLJ-based long-acting drugs, so far, most of the PSGs we recommend in vivo BE studies with PK endpoint. We have one PSG for a dental product that is a locally acting product to recommend comparative clinical endpoint study. And we actually understand and know the challenges associated with that approach. but that was the most viable approach at the time of PSG development. And I think that goes back to another question raised in yesterday's session, why FDA cannot just recommend a PSG with the most sound approach instead of keep revising it. But I think the fact is, when something is complicated, it's complicated for everybody, for industry and for FDA. So it does take everyone's time to really understand and come up with an approach and keep improving upon it. And that was what we have been doing. So for the dental powder product, we have been using that as a model product to study, and we're really getting close to include an alternative in vitro approach. So this slide here just give you an example of PSG. This is with partial AOC because for long-acting products, we often also recommend partial AOC instead of just conventional PK, excuse me, to make sure the PK profiles are comparable, but also the sustainability of a product is also similar. Sorry, excuse me. Then the next scientific challenge or efforts, I would say, we have been working on is the IVIVC. So here I mentioned a couple of times already, totality of evidence in vitro approach. So IVRT is always part of the approach, but because that is a totality of evidence approach, so as far till today, the PSGs for complex LAI products, in which case we recommend in vitro only approach, the IVRT was not targeting IVIVC. So IVIVC is always desired, and it in theory can be a standalone study. But as totality of evidence approach, the IVRT is more targeting to demonstrate understanding of the drug release mechanism instead of really strictly targeting IVIVC conventionally to predict the PK profile. So there is a subtle difference. I'm not discouraging IVIVC, and it can be very beneficial, but just want to point out our totality of evidence, the IVRT, is not necessarily say you have to have an IVIVC. But in order to develop IVIVC, so the paper I'm showing here was published by Dr. Ian Burgess in UConn, and they have lots of experiences exploring IVIVC for complex LAIs. And in recent years, we also start to collaborate and exploring advanced analytical technology to better understand the microstructure of those polymeric systems, so that can inform really the release mechanism. So I'm showing you a few figures here. So we use, for instance, the iron beam focused SEM, and then we take the image and use artificial intelligence informed data segregation to reconstruct the inner structure, and then couple that with our IVRT data to really understand the drug release mechanism. So this slide here, I just give you a quick walkthrough in terms of the timeline of our GDUFA research efforts and how OGD FDA progressed to where we are for the first generic approval. So back in 2013, with the implementation of GDUFA 1, we initiated our first batch of research activities focusing on PLGA-based LAI products. Then in 2015, the first paper published describing analytical methods that can be used to reverse engineering the PLGA-based RLDs. And then in the same year, we revised previously published PSG for risperidone Costa to include specific recommendation on IVRT and further updated our recommendation for the PK study portion. And then the first guidance for octreotide LAR, naltrexone, and risperidone was published or further revised in LDs. Besides the ENDA assessment and our PSGs, then the research outcomes has also been used to address Yeah, I guess the milestone slide wants to have a little bit more highlights. Okay, so the next couple of slides, really I just quickly mention the MVL, which you have heard multiple times. So this is the picture again, and this is a mixture of liposome and microspheres. So the particles themselves are more like microparticles, and they are really in the micron range, and they are sphericals. But then within the spherical, you have this honeycomb structure where the lipids are forming small compartments or chambers. And then that particular packaging has the conventional liposomal feature. So the complexity of this PBPK MVL is a lipid-based microparticle with nano-sized inner structure. It's very complex in terms of manufacturing. We actually, I will show you, we started our internal research, I think back in maybe 2015. And the FDA lab, we found maybe close or more than 30 ways to fail the manufacturing. So that's how many times we have tried and failed. And it's really a big shout out to the lab fellows. Just don't give up until we had a very decent formulation that has this honeycomb structure with reasonable stability. And then this is a locally acting product. Systemic PK is also surgical site dependent. Yeah, then in 2017, we published a paper presenting a very novel IVRT method. Until today, we haven't seen a replicate. What's unique about this method, which made the cover story of Journal of Control Release back then, is it's a testing method coupled with inline UV measurement to continuously monitor the drug release. And we were able to find a condition that really reflects the multiphasic release profile of this MVL product. Its PK profile, as you can read in the labeling, is it has 2 peaks. So it does have a lag phase before the secondary peak or drug is released. Then based on our research, we published our PSG in 2018, and then the first generic was approved earlier this year. But then you can tell from 2018, we didn't really stop there. We know what we recommended in our PSG, and back then we didn't really have all the data to prove the feasibility, or also we want to help generic to better understand our methods. So we had external research carried out reverse engineering and characterization, and we published a paper sharing all those analytical methods. So in today's presentation at the end, I don't have an acknowledgement slide because it's really just not possible for me to have one slide listing all the names, all the individuals that have contributed to the LAI research area and internally and externally. So as a tribute, I'm having 2 slides listing over 40 publications that were produced through the GDUFA research by our internal external collaborators, and also can be a good source for the audience if you are interested in reading some of the papers and learning more about what we have learned. And for my challenge question, it's very simple. Just want to see if everyone thinks all long-acting drug products are considered complex products. True or false? Yeah, it's false because, like I mentioned, the very first generations, the oil solution, those are not complex. Then for drug substance suspensions, they are complex products and they are very challenging to develop as new drugs. But within that, depending on the clinical risk, penicillin G versus paliperidone, we will not consider them at the same complexity level. So what I want to say here is really, when we think something is complex or not, you really have to take this holistic, 360 angle to look at all the aspects rather than fragment and just look at one aspect. So with that, I will summarize. Polymer or lipid-based microparticles, those are very complex, long-acting injectable products. GDUFA research projects have made great contribution to improve the understanding of these products. Those outcomes we have used to develop our product-specific guidances and to address the regulatory inquiries to support the generic development and approval. And OGD will continue to improve PSGs based on updated understanding obtained through communications with generic industry, workshops, CCs, pre-and meeting requests, and GDUFA-funded research. So I think this is really a collaborative effort. We take the first step to publish a PSG to reflect what we think at the moment, maybe the best approach. But that doesn't mean that is truly the best per your perspective. So we really need to hear industry's feedback, criticism, challenges. Those are fine because those are the things push us forward and we should work together and communicate more. So that's why we highly encourage generic industry to really leverage our GDUFA research outcomes and engage with us early during development. So with that, I would like to thank you for your attention and invite all the speakers to the podium. Thank you.
All right, thank you again to our presenters for their wonderful presentations. We'll go ahead and start our question and answer panel to include all our presenters from earlier, and also joining the panel, we have Dr. Hansung Chen, a senior interdisciplinary scientist from the Division of Product Quality Assessment 12, within the Office of Product Quality Assessment 2 from the Office of Pharmaceutical Quality. So thank you again for joining us. As before, attendees in the room are invited to line up at the mics to ask your questions. And then for those online logged in, if you haven't had a chance to submit your question already using that on-screen link and QR code, please do so now. And we'll answer as many questions as we can. So first, we'll see if anyone in the room, again, wants you to come up to the mic. If not, in the meantime, we'll go to our first question online.
Our first question is for Dr. Yang Liu or Dr. Hansung Chen. How was IV-IVC for mesalamine able to capture variability? How many different rates of release formulation were assessed? And can you provide more information on unit impulse response formulation used for deconvolution?
Okay, so can you repeat the second part of the question, please?
Sure, yes. The second part was how many different rates of release formulation were assessed? And then can you provide more information on unit impulse response formulation used for deconvolution?
Yeah, I think maybe Fang is the expert for IVFC, so I'll let her just chime in first.
I think the first question is about the variabilities and also how many immediate delayed release formulations assessed. I think for this one, we focused on the mesalamine delayed release tablets. The ROD is Liada. So we focus on these ROD and the relevant generic drug formulations. We use these as a case example for indicating how PBPK can be used to identify the biopredictive dissolution and predict local drug amount of drug in the colon. So yeah, and regarding the unit pulse for the disposition part, I think they are asking about the disposition. Actually, we use the two-compartment model, and to get the disposition, the PK parameters. The two compartments we have K1 to K21, and also the clearance that we put into the model, and to estimate disposition parameters. I hope that answered the question.
Yeah, thank you, Fang. I'll also chime in a little bit more from the formulation perspective. So for this drug, mesalamine DR tablets, referencing the same RLD product, there have been several endo versions or generic version approved by the FDA. From a formulation perspective, these formulations include the one we have in our case study. They all use very similar excipient, the pH-dependent excipient per se. However, these approved endo products for the pH-dependent excipient they use, they have different amount or they have different ratio. So if we're looking back into the in-house data we have been reviewing for this generic product, we are observing some very different dissolution profiles, either between the RLD and the test or across different endos. So that also reflects the variability of this kind of a complex mechanism of release. However, as Fang just mentioned, these data or this variability or the slight different design of the formulation were taken into consideration when we put the in vitro dissolution data into the PBPK modeling to get a supportive evidence as one of the piece of evidence we use to support either the approval or the decline of the endo.
Thank you so much for all that information provided. We'll go to another online question.
All right, this question is directed towards Dr. Lichen Liang. For products like Teriparatide, which is a polydispersed mixture, and RLD is expected to have a lot of variability, how is sameness comparison performed if the RLD itself is changing?
Thank you for the extremely good question. Actually, I didn't have time to talk about Teriparatide. Teriparatide is a little bit different from the other oligonucleotides because the RLD sequence itself is not defined because it's a mixture of different combinations of sequences. This is by control depolymerization of the DNA from the porcine. So itself has a combination of all different sequences. So instead of trying to compare the sequences, which is very challenging, actually we published guidance and actually advising the industry to actually attack this from different angle. If you use the same source of the the animal tissues or organs and things like that, from the same source, and also use a similar manufacturing process, the same mode of depolymerization, you may have a better chance to have similar kind of distribution on the sequences. So this is quite different from other oligonucleotides. Yeah, thank you. I would highly recommend that you check the PSG for advices. Thanks.
Thank you again for that response. A reminder again to people in the room here, remember, this is your opportunity to get your questions answered right here, right now. So feel free to come up again to the mics. If we don't have any takers for the moment, we'll continue with our online questions, our next one.
All right, we have one for Dr. Yan Wang. Is it acceptable to establish and submit IV/IVC analyses using animal models for LAI products due to the long half-lives for LAIs?
Yeah, thank you for that question. I guess the key in that question is the animal data. So I think as of now, animal data, how we view for generic development, we are certainly open, to get that piece of evidence as like supportive or applicant could use that. And that's what we have been doing in our own research to obtain a baseline understanding of what may be the drug release mechanism and behavior, although species translation is always technically challenging. But as of now, if it's intended for regulatory purposes, I don't think animal data is accepted to support generic assessment or approval. But for development purpose, we are open to look at the data, for instance, in pre-ANDA meeting mechanism and provide feedback to help maybe better design the PK or whatever is relevant to the development.
Thank you again for that response. We'll go to another online question.
Of course, this one is directed towards Dr. Hansong Chen. Is it acceptable to develop a dissolution or IVRT method different from the USP monograph ones?
Thank you for the question. Yeah, this is very general question. The short answer is yes. Yeah, you can develop a different method from the ones listed in the USP monograph. I would like to add a little bit more about this. Our current thinking is, the dissolution method or in vitro drug release method is product specific. So, when you develop the dissolution method, the USP method or FDA database method, it's just the starting point. So you should develop your own method that's appropriate for your drug product. So our expectation is, the method should be sufficiently discriminating against the CMS and the CPPs. So ideally, the method should be clinically relevant. Yeah, thank you.
All right, thank you for that information. Again, still open for anyone in the room to come up if you'd like. But if not, in the meantime, we'll still go to another online question.
All right, we have one for Dr. Fong Wu. For mesalamine, if PBPK modeling shows that dissolution in media less than pH 7 is not as relevant, can the recommendations be changed to remove dissolution testing in these media?
Thank you for the question. So yeah, as we showed that for the Leada and this specific drug product, the dissolution at pH less than 7 appears not be predictive. However, that this dissolution data submitted to the agency still can be considered as some like supporting evidence, or we can also consider look at the data and to see whether there's any difference, and also we consider the release mechanism about these drug product. And then for this product we use the biopredictive solution mainly to put into the PBPK and predict the local drug amount. So we still need that the data at less than 7 for looking at the data. That would I comment. Thank you.
If I may, I also want to provide a little bit comments for that question. So so as I like what Dr. Wang and also Dr. Wu was talking about, and the PSG actually is reflecting the current thinking or the growing understanding on certain aspects based on the knowledge and experience we collected in our review, in our internal research job. Regarding the mesalamine DR tablets, dissolution data under conditions that is a little bit more acidic. So my comment is, so the recommended evaluation stages in the PSG with the 4 different pH values is try to represent all the possible conditions after the drug actually was delivered into the small intestine and after. So this is, you can say this is a more conservative approach. If a test product can have a very similar dissolution in all these conditions, that would be the ideal case. However, just like Fang mentioned, if the applicant observes some issues or some incomparability between the test and the reference in, for example, 6.8 pH condition, it's still recommended to submit the data, and the agency in our review or in the follow-up meeting with the applicant, we are open. Just like the case study we shared today, we are open to consider and take into account other evidence, including but not limited to PBPK modeling, to support and to evaluate the bioequivalence from a totality of evidence perspective. So hopefully that is clear. Thank you.
Thank you so much for that response. And it looks like we have a question in the room. Go ahead.
Yes, thank you, Marie Ducharme, Learn Confirm. It's a follow-up to the previous question that was online. It seemed to me that it made sense that you've proven that 6.8 was not predictive, that you would remove it from the individual product guidance. It seems to me that made sense. Going back to your answer, where you said that the 6.8 was not predictive, here is a question for you. In your PBPK analysis, if I understand correctly, you kind of said that the predictions were validated because you were plus or minus 20% on the mean. And I think I heard that you decided that it was not predictive because when you included the 6.8, you had plus or minus 30%. Okay. My question is, this product is highly variable, right? Correct? It has 70% to 100% intra-individual variability. Correct? So why would plus or minus 30% not be good when it's highly variable at plus or minus 70 to 100%?
Yeah, so first I would like to clarify, and just now we said that although we showed that the solution at pH 6.8 is not biopredictive, but this is still included in the current PSG. And also we are currently thinking is that data can still be submitted to the agency, and we are going to look at it for evidence and considering the study, the formulation design, release mechanism.
I understood that.
Yeah,
I understood
so
that.
just not to be removed from the PSG, just to clarify that. And then for the variability you mentioned, yes, the mesalamine, it is a highly variable drug product. And what we generally consider, which is biopredictive, is based on our current practice on using PBPK modeling, especially for the BE assessment. If the prediction error is more than 20 or 25%, that considered is not a good prediction. So that based on that, we considered that the pH 6.8 included that is not biopredictive as compared to the observed PK. Yeah, and yeah, I understand.
I'm sorry, but in previous presentation by the agency, validation of PBPK is usually plus or minus twofold.
So yeah, I think
I'm really surprised that you're accepting plus or minus 20% and then rejecting plus or minus 30% when this is a product that's highly variable. I mean, it's a little bit surprising.
Yeah, it's not rejecting, just comparing to the other dissolution that they are.
But that's how you decided that 6.8% was not, that 6.8 was not relevant. That's how you decided that, because you got plus or minus 30%. I mean, think about it. I just, anyway, a comment for thinking.
Yeah, thank you. Thank you for the comment. Yeah, for the twofold, actually, we are considering whether it is validated or not, depending on the modeling purpose. The twofold, yeah, as you know that the DDI, when we use the modeling to see whether it is validated or not, sometimes twofold we can consider, and also if there is a the platform validation, we can consider that big variability, the prediction error. But for the PE assessment, generally we consider like 20%, around 20%. Yeah, thank you.
Thank you for that. We have another question here. Yep.
So I have a question for the PLG, the long-acting drug. So the PLGA. The PLGA is an excipient, and there's a lot of study, a lot of focus from a talk. So from a previous talks, especially yesterday, it's API has been focused a lot. Your talk, PLJ, has a lot of research started there. So my question is, what reason behind that? Just you want to make sure the release duration is okay, or you also talk about impurity or something else? Why that has so much focus?
So your question is why we spend so much efforts on characterizing the PLG, considering it's being
just
an
excipient.
excipient?
Yep.
Okay. Yeah, I think that's a fair question. So first of all, PLG is the excipient and it's actually a functional excipient, meaning its characteristics plays a very important role for the drug product performance. Then the main reason for us to spend so much efforts on understanding the polymer, one part is because its impact on the product performance, then that directly relates to the successful rate of having a successful product. But the other hand, as I mentioned, is to fulfill our regulation. So the regulation, because the PLJ-based products, most of them are parenteral products. So the regulation requires the generic to demonstrate the composition of the generic is qualitatively and quantitatively the same. But then for PLJ polymer, because of the inherent variability, is not as straightforward as some excipients. Q1, it strictly goes to chemical identity. So our efforts is really to understand the characteristics of the polymer and the source impact of the polymer on the product performance, and also to guide. Right now, we have made great progress in terms of we have clear guidance to tell the applicant what they should look for, what they should do. But then having said that, we also understand from generic development standpoint, it's still very restrictive in terms of today, probably in terms of characteristics, the molecular weight, we have probably the max flexibility depending on the product and our understanding. So if we know a product, the PK and its release is not so sensitive to differences in molecular weight, mean molecular weight and distribution, then on face value, if we see a 10 to even 20 KD difference in the mean molecular weight, we would still consider that as the same polymer. Then, but in some other cases, depending on the product or dosage form, we also know if you have less than 5 KD difference, you will see a difference in the release and the PK. Those things may not be obvious to applicant depending on how much experience they really have. And that's why we do those research and publish, share our understanding, and also we provide as much detail recommendations in our responses to their inquiries to facilitate their development, to improve the successful rate, because those are so complex and costly to develop, they probably don't have many chances for failure.
Thank you. I guess we can talk a lot on that in offline.
Yeah, sure.
All right, thank you again for that response. Maybe we'll try to fit in one more online question.
Sure. So we have a question for Dr. Yang Wang. So are there any validated mathematical models for establishing IV-IVC for long-acting injectables?
A very good and challenging question. As of now, I think the IV-IVC we have successfully demonstrated in animal, those are more empirical mathematical model. So either time shifting or time factoring based on mostly, the release mechanism. If it's sigmoidal triphasic release, we would try to keep the shape of the in vitro, then just compare the time scale and factor that. Having said that, there are ongoing efforts actually in the sister division, DKMM, to look into PBPK, for instance, those model to establish IV-IVC mechanistic modeling. And also from our perspective, like I mentioned briefly, we are also looking to like the physics, the AI-based simulation, and then the imaging microstructure supported release mechanism exploration. And we are looking to using those tools to establish IV-IVC. But at the moment, I don't think we have a robust, validated mathematical model that can fit all products.
All right, thank you again for that response. And I know we're going a bit into lunch break, so maybe we'll go ahead and end our session there. But thank you again to our presenters and panelists for all the excellent information and insights. Let's go ahead and give them another hand. Yep. All right. Okay, and so our plan will be to resume again at 2 p.m. For those of you here in the room, we have a few additional announcements. Just a reminder that we have some lovely poster presentations or prepared out there by our subject matter experts on display. So this is a great time to take a look at them if you haven't already. Also some news on another food option. There is a lunch buffet being offered downstairs for $20 right here on site downstairs. So it's something for you to consider. And then additionally, there is a parking discount available for attendees for you to only pay $10 for the day. So if you haven't gotten information on that already, please see the team at the registration table. All right, and so we'll see you back at 2 p.m. Right.
So the podium is yours.
All right, thank you. All right, everybody, we're going to get started again. So welcome back from the break. Hope everyone had a great lunch or got the time to enjoy whatever you needed or wanted to do. We're in the home stretch here, so thank you to all of those who have made it back after the break. To keep things a little interesting, we have a quick fun activity with a potential prize bag on the line here for those in the room. Just curious, has anyone heard of the SBIA podcast? Could I get a show of hands of anyone who's listened to it? Okay, awesome. So the question for today is, does anyone happen to know what the name of the podcast is, or the name of the newsletter that we've reformatted as the podcast? If you happen to know it, you can just call it out. Did someone say it over there? No? Okay, no worries. So we can go ahead and do our reveal. So it's actually the SBIA Chronicles podcast. So this is a newsletter that's we've read out into audio format, so you can listen to it on the go. It's available on different platforms where you can stream it. If you could scroll down just a little bit. So, oh, that's okay. But yeah, I just wanted to show it's available on different podcasts there, wherever you may choose to find it. And that way you can get your updates on the go. So thank you. We'll go ahead that. All right. And so now it's my pleasure to introduce our speakers for our final session here on the that's going to be on ensuring efficient and consistent high-quality generic drug development. Okay, so please join me in welcoming our presenters. First, we'll hear from Dr. Greg Huang, a senior chemist in the Division of Product Quality Assessment, or DPQA 9, that's within the Office of Product Quality Assessment, or OPQA. And then we'll hear from Dr. Feng Liu, a lead pharmacologist in the Division of Bioequivalence 1 within the Office of Bioequivalence in the Office of Generic Drugs. Then we'll hear from Dr. Priyanka Ghosh, a lead pharmacologist within the Division of Therapeutic Performance 1 in the Office of Research and Standards within the Office of Generic Drugs, followed by Dr. Lei Zhang, Deputy Director of the Office of Research and Standards within the Office of Generic Drugs. And then we'll also hear from Dr. Yuqing Gong, a senior pharmacologist within the Division of Quantitative Methods and Modeling, or DQMM. And that's also within the Office of Research and Standards within the Office of Generic Drugs. So now I'll hand it over to our first presenter, Dr. Greg Huang.
Hi, I'm going to talk about the guidance for industry content and the format of composition statement and the corresponding statement of ingredients in the labeling in NDAs and ANDAs. Even though it is about a policy, today I'm more focusing on the CMC perspective regarding the ANDA and the NDA submissions, what should be adequate information to be included in a composition and the labeling. So today's learning objective, you will be able to describe content and the format in the composition statement of a drug product. You'll be able to describe what exactly to include in the statement of ingredients in the labeling. And you will be better understand the requirement for consistency in composition information through the NDA and the ANDA submissions. So the FDC Act and the 21 CFR 314 their requirements for NDA and ANDA to provide full list of components used in the drug product manufacturing, and also provide a clear statement of composition in the drug product. So this guidance is a draft guidance. It is fairly new, published 5 months ago in April, first draft. So this guidance recommends what kind of accurate and complete information regarding the composition statement and the statement of labeling, ingredients in labeling should be provided. This will ensure that FDA approved labeling and composition contain adequate qualitative and quantitative information. And this definitely will help a CMC assessment to ensure we have an efficient assessment process and minimize the communication cycles and lead to fast approval of and NDAs. So what exactly we mean by composition statement? So it should include the qualitative and quantitative information of active and inactive ingredients of a drug product. Specifically in the ECTD submission is in module 3, section P1. And what is the statement of ingredients? It means the information of all the ingredients in active and active listed in the labeling, which is in the ECTD module 14, and including other types of FDA-approved labeling, such as the cartoon container labeling, patient insert labeling. So what should be included in the composition statement? So first of all, first is the name of all the active and inactive ingredients with clear identification, such as grade and type. So for this, you can refer to the established name of USP NF monograph, and also the FDA Global Substance Registration System, or GSRS, to refer to these source. And for the GSRS, the ingredients has a unique ingredient identifier, or called a Unicode. So it also, second component of the statement should include the reference, compendial reference standard. If it's a USP/NF, then it should be clarified. If it's in-house, then it's in-house. And also the primary function of inactive ingredients, which means the role of the ingredients played in the formulation. You can further refer to the USP General Chapter 1059 for the excipient performance for the functional categories of the excipients for further information. It should be a straightforward terminology, such as preservatives, antioxidants, buffering agent, or salt-forming agent, etc. should be clear. So the fourth component is the quantity of each ingredient. It includes the concentration in the liquid, mg per ml. Mg per ml, and also the amount or strength per unit. And also it can be the percentage by volume or weight. And it should be in the same units for each quantity category. So here's an example of the composition statement. So this is an example for injectable injection drug product. Sodium Jugazide is a hypothetical API for the purpose of a presentation. So it includes name So the name is very clear and specific. Either it's a API sodium salt, or it's a sodium citrate dihydrate, or a citric acid monohydrate, edited sodium dihydrate. Is very clear. Okay. Second component is about the material related to the compendial standard. So in this case, they are all USP compendial. And the function is API, tonicity, buffer agent, preservative, and the quantity in mg per mil, mg per vial unit, and the percentage of weight by volume. And this is continued example for the name. It's very clear, specific, povidone K17. And also the compendial standards, it's could be in-house and USP or national formulary or in-house. Yeah, the function and the quantity. So this is a complete and accurate representation of the formulation. So I'm going to give a little bit more specifics how to identify ingredients in composition statement. First of all, the active ingredients, it can be in a salt form, even though the potency in the labeling typically is expressed in the potency of the free base or active moiety. So, both of the information should be clarified in the composition statement. And if API is a mixture, so it can be a chemical isomer or sterol isomer, then it should be stated clearly, and the composition. If it's a racemic mixture, it also should be clarified. So for inactive ingredients, hydration form, the grade composition of the ingredient, and a type of polymer, it can be molecular weight or the different composition. We are going to give a more example in later slides. And also purity. So I'm going to give an example to identify API in the module 3, P1. P1 is the description and the composition of drug product. So here it says sodium jugoxide is a salt of jugoxide converted by reacting with sodium hydroxide. Each ml of the drug product contains 27.3 mg sodium jugoxide, which is equivalent to jugoxide, which is a free base of 25 mg. So in this case, we assume assume the API is a mixture. So you would say jugoxide is a mixture of jugoxide A and jugoxide B in a ratio of 3:1. This is an example to explain it. So yeah, this is very clear. So about the inactive ingredients, what information to include? First of all is the hydration form. So in this case, the sodium citrate, citric acid, added sodium. So they are differentiated by anhydrous, monohydrate, or dihydrate, other forms. I know the USP monograph may cover different hydration forms. So, but the recommendation is to be clear what form you are using in the formulation. So the next one is a polymer type like povidone. The same USP monograph can cover different nominal K values. And the second example is hypromellose. Differentiated by substitution type of methoxy and hydroxy propoxy percentage. The next one is hydroxyethyl cellulose, which contain different proportion of hydroxy ethoxy group. And the carboma has a different type by the cross-linking and copolymer composition. More example about the composition Like a stearic acid, it can be differentiated by the percentage of stearic acid and palmitic acid. And also the next example is about the purity accountant, acetic acid and glacial acetic acid. They're differentiated by the different purity, and alcohol and dehydrated alcohol, also the same, differentiated by the purity. So more specific about the statement of ingredients in the labeling. So this means the list of ingredients in module, ECTD module 1, section 14, it should be consistent with the statement of composition in module 3, P1. And qualitative information for all the active and inactive ingredients in the labeling, and a quantitative information of API for all drug products, and a quantitative information for inactive ingredients for parenteral injection. So here is an example. Again, it's for a hypothetical injection drug product. So the description of labeling in ECTD 1.14, it says each milliliter contains a drug dose, drugazide 25 mg, equivalent to the sodium salt 27.3 mg, and the following ingredients: citric acid monohydrate 1 milligram, etc. So they are specific in the type of the ingredients and the amount, and also the pH adjuster. It does not, if it's a use adjuster, it does not have to be quantitative information provided, but definitely it should be listed in the labeling. So some observations during our CMC review, and this first one is observation. Second is how it should be clarified. So first, a compendial reference not identified. So it should be compendial or in-house. And hydration form not identified. Very common for us, like sodium, added sodium, and just added sodium. We don't know what kind of form it is. And sodium carbonate monohydrate, so it is clear on the right column. And the pH adjusting agent not listed or clarified. It's not in the labeling, or it can be missing from the composition statement also. So it should be clearly listed. And inconsistent unit of concentration for inactive ingredients. So sometimes all the ingredients are expressed in percentage weight by volume, but one of them may be presented in a different unit. This is not a good situation, so it should be corrected. And for the conversion, you may also provide additional information such as density and the temperature, etc., to convert to a consistent unit of ingredients. Okay, so another example is a composition of active ingredients deviated from the USP monograph. So in the case, the raw material may be pre-processed. So in this case, is a pre-acidified polysorbate 80. So it's considered in-house, even though your starting material, polysorbate 80, is a USP NF grade, but this is pre-processed. And the grade of the inactive ingredients are not clarified. So on the right column, the clear example is povidone 17 or hypromellose 1822. They are very clearly specified. So we just talked about the composition statement, how we should include in terms of content format, and also the labeling, what should be included in the statement of ingredients. Throughout the CMC submission, actually there are more to be taken care of. First of all is the in the batch formula in the module 3P, section P3, and the master batch record, executive batch records in the relevant modules, they should be consistent. Sometimes we see inconsistency, and this should be noted. So also, if the multiple manufacturing facilities include, there may be some variation. Okay, so the composition statement and the labeling should include all possible ingredients and levels in the composition statement, and be consistent in the labeling. So here's a few examples for inconsistency we observe in the NDA and the ANDA submissions. First example is acid base added as pH adjusters, but they are omitted either in the P1 composition of the drug product, or they could be omitted in the batch formula. So we needed to make sure that the composition table in P1 and the batch formula include consistent information about the pH adjuster. So the second example is a composition table lists the ingredient in fixed amount. However, in the batch record, it says QS. Okay, this is not consistent. So they should be consistently presenting each active and inactive ingredients and their amount. So with all that, I want to present a challenge question. First is the following should be included in the composition statement of a drug product. First, name of all the active ingredients and inactive ingredients. B, a reference quality standard for all ingredients. C, primary function of active ingredients. D, quantitative information. And E, all. Which one? Oh, I heard all. I think all of you. Great. Okay. Second question, which is not needed in the statement of ingredients in labeling? This could be more than one answer, not sure. First, name of all active and inactive ingredients. And the pH adjusters, grade or type of inactive ingredients, polymorphic form of API, processing aid used in the drug product and manufacturing. Which one? Which one, D? D and E, okay, great. Yeah, so I should emphasize this is still the very first draft. Is still revising, and we are addressing public comments. So we definitely welcome more questions in the panel discussion. Actually, my colleague Rachel is a panelist, so we can discuss, and also refer to the 1059 for excipient performance as a resource. So in summary, composition statement should include clearly identified ingredients and their quantitative amounts. Statement of ingredients in labeling should list complete ingredients that are consistent with composition statement. And batch formula and manufacturing batch records in the ECTD submission should be consistent with composition statement. So I'd like to acknowledge my colleagues at Work Unit OPQ2 of OPQ for their feedback, and also our wonderful guidance working group when I prepared this talk. I got very good feedback, and I thank all of them. So next, I will invite Dr. Fang Lu to the podium.
Good afternoon, everyone. I will share with you the analysis of the first cycle BX rate and major BE deficiency encountered from bioequivalence perspective. Self-introduction. I'm a team leader from Division Bioequivalence 1, OB, OGD. Learning by sharing. When I prepare this presentation, 2 questions pop up in my mind. Number 1, what is the first cycle B efficacy rate? Number 2, how we can improve first cycle B efficacy? To answer those 2 critical questions, I will present the information as outlined here. Number 1, purpose. Number 2, data-driven outcome. Number 3, tips that could improve first cycle B efficacy. Number 4, summary. The more review cycle during the review process, the longer time to achieve B efficacy. Therefore, we'd like to share with you first cycle B efficacy and the major BE deficiency type encountered, along with those suggestions how to avoid those deficiencies. We retrospectively assess the submission quality and commonly observed BE deficiency from 729 ANDAs submitted during GDUFA III Year 1 and identify 82 ANDAs are BE inadequate. One review cycle is defined as FDA received the application to the first CR letter with either BE inadequate or BE adequate outcomes. The first cycle B efficacy rate is 87% for 335 ANDAs assessed by OB. Out of 82 BE inadequate ANDAs, 57 and 43% were classified as a BE major or minor deficiency, respectively. All right, it's a busy slide. However, we'd like to present the BE major deficiency themes from an overview perspective and summarize as a 7+1 top themes as in this graphic. Most of the deficiency themes are inadequate in vitro or in vivo B studies and method validation as well. We also identify the impact from other disciplines, include data integrity issue, identified by oath inspection, or the batch not acceptable as determined by OPQ, or exhibiting with a safety concern by PharmTalk. We will further explain the major B deficiency by using case scenario with tips how to avoid these pitfalls. For example, in case number 1, the statistical result provided by applicant only include 2 decimal places, where as our BE assessor found that the study failed when conducted confirmatory calculation applying 4 decimal places as a specific recommend in guidance. Please note that all in vivo B study and in vitro dissolution testing data are considered invalid once the biobatch has not been found acceptable due to many reasons, such as the API source has a CGMP issue, or drug products quality or stability issue. We found one case that the capsule stick together became too yellowish during the storage, so the batch is not acceptable. So all of the study considered invalid. So please see the tips here. Wisely select API suppliers. Ensure your drug product's quality and stability, and maintain the manufacturer site compliance. In this table, we summarize each BE major deficiency themes, followed by sample deficiency with the tips. For example, when exclude a subject data under product deviation, we suggest to submit a full investigation report from both bioanalytical or clinical deviation perspective. The key point is here with the real-time evidence, including your statistical plan. For those easily correctable deficiency, we suggest that before submission, carefully check if BE study report or data are complete and consistent as possible. For example, double check if your SAS data set submit as a CDX format. In many cases, you can avoid easy correctable deficiency. PSG is a useful resource to avoid those BE deficiencies. For example, original PSG include only one in vivo options. However, the revised PSG including both of in vitro and in vivo options. So you have more options. For those complex drug products with a challenging issue, we encourage you to discuss with us on your BE strategy before submission by appropriate approach. Of course, you may also propose alternative in vitro or any in vivo approach with a scientific justification. So here we have some so-called challenging question, but I think simple question here. Number one, the first cycle BE adequacy rate. The answer is simple, C. So this a little bit challenging. In which of the following scenario could a biobatch still be considered acceptable? I better give you the answer quickly, okay? Age batch with appropriate stability, the rest of unacceptable. So in summary, the data-driven outcome indicate that first cycle BE adequacy rate is about 87% for 635 ANDAs assessed by OB. Please consider those tips in slides number A to 12, not K to 12, in the presentation. Let's work together. For example, communicate with us by appropriate approach on your BE strategy to bring effective, safe genetic drug products to American people. I appreciate my OB colleagues, especially Utpal, Tao, Chicha, Sri, Zen, Bing, and Pasa, for their help on this presentation. Of course, I appreciate all OB assessors and project managers. Without them, the data cannot be collected. Thank you. Let me turn this stage to Priyanka.
Okay. I'm going to continue the discussions related to ANDA approval and major deficiencies. And I'm going to use a case study with topical products for the purposes of this presentation today. So the learning objectives for us is to understand the ANDA landscape for topical products applied to the skin, specifically what kind of approaches are being utilized to support an evaluation of BE. And then to understand what the challenges are and how do we resolve those challenges so we can eventually get to first cycle approvals. So the outline of my presentation has a brief recap of the BE approaches that we currently recommend within product-specific guidances for topical products. And then the second part of the presentation talks about landscape analysis of ANDAs that have been submitted to the agency since GDUFA II, so FY18. And then, of course, the challenges and opportunities. So a brief recap from the presentation that Dr. Kelchen provided yesterday morning. There are predominantly 4 types of BE studies that are currently recommended within the guidances for topical products. We have the comparative clinical endpoint BE studies. These can be used by all products irrespective of the formulation. Then we have the vasoconstrictor studies. These are typically used by corticosteroid products. We have situations where a waiver of in vivo BE studies may be applicable, typically for topical solutions where bioavailability may be self-evident. And lastly, the characterization-based approaches that have been developed within the scope of the GDUFA Regulatory Science and Research Program. So let's now look at some data. So I'm going to take some time on the slide to orient you to the slide because all of my subsequent slides are structured the same way. This is a summary of the submitted ANDAs that we have received since FY18. And the way the data is presented here is data for a given fiscal year is normalized to the total number of ANDAs received in that fiscal year. For the purposes of this presentation, approved ANDAs indicate any ANDA that has been approved or tentatively approved. Pending ANDAs indicate any ANDA that is in pending status, either in a CR status with the applicant or in pending status with the agency, and of course, withdrawn ANDAs. So overall, if we look at all of the data that we have since FY18 till FY24, the data is as of June 30th of 2024, we see about 55 to 57% of the ANDAs that we have received during this time for topical products applied to the skin have already been approved. Now let's take a look at what kind of approaches were used to support BE in these submitted ANDAs. So if we focus on the left of the screen here, we see the characterization-based approaches. And since the publication of the guidances related to these characterization-based approaches in the FY18-19 timeframe, we have seen a reasonable increase in the number of ANDAs that have utilized characterization-based approaches to support BE. And the number that I would like for you to keep in mind is this FY2021 number, where about 70% of the ANDAs used the characterization-based approach. We have also seen consistent submissions for products which have used clinical endpoint BE studies, VC studies when appropriate, and of course waivers when appropriate. Now let's look at what has been approved. So when we look at approvals, if we focus on the characterization-based approach first, remember the number 70% that I mentioned for FY21, we see that about 35 to 40% of the ANDAs have been already approved. So overall, we see approximately 40% of the ANDAs that have used characterization-based approaches have been approved until now. We also see consistent approvals for the clinical endpoint and the VC studies, but I'm going to talk about those in a little bit more detail when I talk about pending ANDAs. If we take a closer look at the approaches that used characterization-based approaches, we see that a substantial number of those products have successfully completed IVRT studies, and also some of those products have successfully completed IVPT studies. So I just wanted to note that before we look at the challenges we face in this area. So now looking at pending ANDAs. As we look at pending ANDAs in trend with the submissions where we saw an increase in utilization of the characterization-based approaches, we also see an increase in the number of pending ANDAs when a characterization-based approach has been used, and I'll delve into this a little bit more on subsequent slides. For the clinical endpoint studies, VC studies, and waivers, we do not see as many pending ANDAs, but I'll talk about overall some of the challenges that we do observe. So for the overall pending ANDAs, if we were to break them down about the challenges, we see about 50% of these products still have outstanding challenges related to bioequivalence, pharmaceutical equivalence, and in some cases also labeling, for example. But the other 50% of the ANDAs where bioequivalents have been found to be adequate, there are outstanding challenges related to pharmaceutical quality. They could be related to either drug substance or drug product, and also related to manufacturing and process and the facility. A limited number of cases where we have labeling challenges. So one thing that I wanted to note here is, while the pre-NDAs that I have been involved in have predominantly focused on the BE approaches, because this is new in the area of topical products, there may be some room for discussing pharmaceutical quality-related challenges in the pre-NDA space when those are appropriate as well, so that we can resolve those challenges and get to that first cycle approval. Delving down a little bit into these characterization pending ANDAs that utilize characterization-based approach, we see about a quarter of these ANDAs, a quarter to 30% of these ANDAs have adequate IVRT study. For the remaining ones, while this segment looks big, I will note that the majority of the challenges that we observe with IVRT are primarily related to IVRT method validation or analytical method validation. And with the resources we have currently available, guidance, trainings, I think a majority of those challenges are resolvable within one cycle. With IVPT, however, we see that approximately 79% of the ANDAs that we have pending and which require an IVPT, the IVPT is inadequate. So this is a significant challenge for us. And when it comes to IVPT, we have observed that there are challenges related to method development. And what I mean by that is we observe that there is inadequate optimization, for example, with respect to the receptor solution. It is extremely important during IVPT method development to optimize the receptor solution for a given drug molecule, a given drug product, to ensure that there is sufficient sensitivity, sufficient solubility and stability of the molecule in receptor solution. And it is often not appropriate to just use a receptor solution that is available in the lab and proceed to method validation and pivotal studies. Another example of a challenge that we observe is with sampling frequency. We often see methods that are being submitted where a standard sampling frequency is utilized without any consideration for how that sampling frequency impacts the shape of that permeation profile, and if that sampling frequency allows us to truly compare two products. So one potential option to resolve some of these challenges, because we do recognize there are challenges with IVPT method development, is to potentially discuss with us as you are developing your IVPT method, as you are running into these challenges through the PNDA path, and specifically through the product development meetings. The other major challenge that we observe related to IVPT is related to method validation. And there we see challenges related to the selection of the dose, for example, for the IVPT sensitivity study, or the selection of the alternative formulation for the IVPT selectivity study. And in this situation also, we feel like we have guidance out there about how to select the dose. We have presentations out there about how to select a dose that will give you the separation of the profile between your target dose and your selected dose to be able to establish the sensitivity of the IVPT method. But we also recognize that since assessing sensitivity and selectivity is a qualitative assessment at the moment for our guidances, this can lead to some challenges. So again, as you are developing your IVPT method, as you are validating your IVPT method before you proceed to that expensive pivotal IVPT study, if you have questions, we strongly encourage you to interact with us through either the product development program, or in some situations, even the pre-submission program may be appropriate. There are, of course, other issues related to aberrant data in IVPT studies. And given all of these challenges that we are observing, we hope to host a CRCG workshop in March of next year to address, to discuss a lot of these challenges in a lot more detail. So in conclusion, I would like to say that the landscape analysis of the ANDAs in the topical product generally indicate that characterization-based approaches have been predominantly used to support ANDA submissions since GDUFA II. And overall, not just using characterization-based approaches, we have approved over 175 topical ANDAs since FY18. Among the pending ANDAs, there are challenges related to pharmaceutical quality, labeling, and/or BE. However, for the products where characterization-based BE approaches were used, the predominant challenge appears to be related to the IVPT studies. So we would like to, again, recommend engagement with agency early during product development, as you are developing your methods, as you are validating your methods, so we can resolve some of these challenges and support product approval. So with that, my challenge question, what are the pathways that can be used to interact with agency? A product development meeting, a pre-submission meeting, a post-complete response letter related meeting, controlled correspondences. I would say you can use all of the above, whichever seems most appropriate for your specific question. So with that, I would like to acknowledge my entire team within the Office of Research and Standards, and I would like to invite Dr. Lei Zhang for her presentation related to ICH M13.
Okay, good afternoon. Next, I'm going to give an update on the ICH M13A, which is the first ICH guideline for bioequivalence. And you may be aware from yesterday's opening remarks from Commissioner or from the news, this really represents the milestone for the generic drugs in terms of BE in the global level. So I will provide an overview of the M13 guideline series, not just M13A, but what's the plan, and also provide an overview of the final M13A guidance and major changes from the draft, and also what the major difference from the FDA current general guidance, as well as provide an update on the FDA planning on the implementation of M13A for the generic drug development. So for this audience, you all aware the bioequivalence assessment is really very important for the establishment of the therapeutic equivalence for generic drugs to their respective reference list drug. And also you may be aware the reference list drug is the terminology FDA is using, but for the M13A, comparator product was utilized. It's equivalent to FDA's RLD, which represent the standards, this is the terminology we harmonize at the global level to represent the reference standard other regulated agencies will recognize for their regulation needs. And we also know BE is not only for generic drugs, they also be utilized by new drugs for their decision-making, either pre-approval or sometimes the post-marketing, post-approval formulation or manufacture changes. So it's very important a concept. However, traditionally, we know in the ICH level, there's no harmonized guidance for bioequivalence or for generic drugs. So this is really the first topic bring to the ICH. Starting with the reflection paper that was adopted by ICH in 2018 on further opportunities for harmonization of standards for generic drugs, which laid out a strategic approach on how we harmonize and provide these scientific technical standards for BE, starting from a more simpler product to more complex products. And the ICH also formed the generic drug discussion group following the reflection paper. The discussion group really have representations from not just the regulatory agency and the ICH, but also the industry groups to recommend topics for harmonization and what should be the order for prioritization. And they also supported the submission of the first new topic proposal, which is lead to the M13 expert work group formation, focus on the immediate release oral solid dosage form. And the M13 expert working group was formed since 2020 and mainly focused on immediate release solid oral dosage form. And based on the scope of the work, the EWG proposed to develop M13 in 3 series. So M13A really represents the first guideline under this series focusing on the B study design and analysis for immediate release solid oral dosage form. And later there will be a M13B, is second in the series, will cover the topics on additional strength, as well as including the additional strength waiver. And the M13C will cover the data analysis and the BE for highly variable drugs, therapeutic drugs, as well as some more complex study design and data analysis, including adaptive design. The reason to break this into 3 series, so the working group will be able to break down the topics so we can deliver those finalized guidance in a timely manner, in sequential, but also consider there could be some overlapping while we work on those guidances in order to bring the efficiency. So right now the M13A is finalized. We are very happy. Now it's reached the step 5, and it's ready for the global regulatory agency to implement this guidance in the generic drug development. M13B is ongoing, and the working groups currently work on reaching consensus on the draft technical guidance to anticipate to reach step 1 towards the end of this year. And the M13C has not started yet. It will start after M13B is the draft is published and during the public consultation stage. So it is really a long journey when I'm looking back. I have the privilege to serve as the rapporteur for the M13 expert working group. And Nilufer Temple, who will be on the panel, she is the FDA topic lead. We have a very diligent and collaborative global working group under ICH with 22 members, observers, as well as among them, 14 of them are the regulatory agencies. So we have been working very hard in the past 4 years by published this guideline, follow the ICH process for harmonization. Really, there are 5 steps as laid out in this slide. And we delivered the draft guidance towards the end of 2022, and we received a lot of public comments, and we reviewed all of those comments and the finalized guidelines based on the comments received and understand the areas that may need additional clarification. And the final guidance was finished and adopted by the ICH in July of this year. And now it's ready for, at step 5, ready for implementation. And the global regulatory agency under ICH also have their own plan for the implementation. And the FDA is also in the process of revise our general guidance as well as the product-specific guidance to align with M13A. So the overall content of M13A in the final guidance does not change from the draft guideline. There's 5 sessions, including the introduction that will cover the objectives, background, and scope. And the key content is in the section 2, which talks about general principles for establishing BE with both study design and the data analysis part. And we focus on non-replicated study design. And we also have 4 sections covers various specific topics on endogenous compounds, other immediate release dosage forms, which including the oral disintegrate tablets, chewable tablets, and oral suspension, as well as the fixed dose combination. And we also have the session talk about the pH dependency, when you may need to consider one additional study under the with a acid-reducing agent in order to establish BE. And we also have a section on documentation and glossary. So during the public comment set period, we received close to 800 non-replicate comments, and the majority of them are related to Section 2. It's not surprising because this is the key part of the guidance. We have 77% comments related to that section. In addition, we have 10% of comments also related to Section 3. And the major comment themes are focusing on, because we introduced the risk-based approach for determining whether we need 1B studies or 2B studies under what fasting or fed conditions. So there's a lot of clarification needed on how you define high risk and when fed B study is needed. We also received some other comments related to how you define the dose proportionality. This is also related to M13B in terms of additional strength waiver, and also study population, data analysis, various aspects of it, group effect, and also alignment of study design with product labeling. So the expert working group review all the comments, and the modification are being made to based on those comments received. And also, in addition, we also realized some topics may need additional clarification or rationale, which we don't have the space in the guidance to provide that. So when we finalize the guidance, the working group also developed a questions and answer document. It's published the same time as the final guidance. So we don't think there's a major changes from the in terms of the recommendations from the draft guidance, but we did make modifications on several sections, mainly to provide clarification to help the implementation or make them more practical for the regular agency to implement the guidance. So in particular, the high-risk product, what does the risk mean in this under the context of M13A? It is really to mean the risk of the B inequivalence or B failure for those products which has special characteristics. They may result in a high risk of the B inequivalence under the fat condition due to the food effect, differential food effect caused by formulation or manufacturing process. And also the data analysis part has some more clarification provided, as well as PK dose proportionality assessment, early exposure. We, in addition to the partial UC, we also introduced the Tmax may be used, and also additional clarity on pH dependency when you may need an additional study, and what justification you may provide. So as I mentioned, the working group provide additional questions and answers document to help provide further clarity and also assist the implementation. As you can see, all the Q&A are focusing on section 2, 3, and 4. In addition, as part of the ICH process, a step 4 presentation was published the same time. This is serving as the basis for training materials to help the implementation, and the links on the slide. So if you read the guidance, before the M13A, the different agencies do have, even the B has been used for many years. The regular agency globally do have different recommendations for B study design and the criteria to support the generic drug approval. This may result in inefficient drug development, increased cost and time. Additionally, maybe cause some drugs may not be approved in certain market. And the major difference we are aware is the FDA generally recommended both fasting and fat studies. However, several other regular agencies, including the European Medicines Agency, EMA, they generally recommend one study, which is fasting studies. So those differences in the general B guidance can lead to the different product-specific guidance recommendations, which both FDA and EMA has PSGs developed in addition to the general guidance. So this table just really to show you those differences in the general guidance does translate to the PSG recommendation. If you look at the comparison between US FDA and the EMA, in terms of PSG, although FDA do develop more PSGs than the EMA, but if you look at the percentage-wise, we do have a majority, like 86% PSG prior to M13A recommend both fast and fast studies, while EMA only have 16%. And they have majority recommend the fasting studies only, because this is reflected by the general B guidance differences. So as we are aware, the M13A introduced the risk-based approach to determine the need for the B study condition. So for high-risk product, as I briefly alluded to, those are the products which difference in the formulation or manufacture process may not be detected with a single B study, either in the fasting or fat study. Fat condition may not be extrapolated to predict B under other conditions. Therefore, the guidance recommend the B study for those type of product, B should be conducted in both fast and fat conditions, and also Q&A touched further rationale on why we recommend this. And for non-high-risk products, on the other hand, the results from a single study, which typically could be a fasting study, maybe could be extrapolated to predict Fed B study outcome or vice versa. So the B test only one condition is sufficient. And the guidance, the revision, you can see there's some slightly different language used in the final guidance, but the concept is not really changed, but we just try to articulate better how you identify a product as a high risk, non-high risk, what you may consider when you develop your product to consider whether you need to do 2 studies versus 1 studies. So really we need to look at the reference product or ILDs, drug substance characteristics, coupled with their formulation, design, or manufacture process, and what's the labeling used, what's the food effect, how they those with relation to food, and their specific product formulation that consider high risk, which a couple of examples were given in the guidance, which include the solid dispersions, microemulsions, lipid-based formulation, nanotechnology, and some other specialized technology. And also in the guidance also alluded that the test product also need to consider how you, similar to the reference product, just because some product or some reference product may not deemed to be high risk. However, although some product may not use the complex formulation, but the specific characteristics in the formulation, if it's different between the test and the reference product, they can result in a formulation that may modulate the food effect different, and there might be a need to conduct a study under both conditions or provide justification. And also in the guidance, both in the draft and also in the final guidance, talk about the meal type for Fed B studies. So for high-risk product, the guidance recommend in respective of product labeling, the study need to conduct both fast and fat, and the fat, it will be the high-fat, high-calorie meal. However, for the non-high-risk product, if the Fed B study is needed, especially when that's where the labeling indicates the drug should be taken with the food for a PK reason, then either a high-fat, high-calorie meal or low-fat, low-calorie meal can be used, administered, and also labeling. And also if the labeling specifies certain meal, that could be the meal used for the B studies. And also the additional rationale will provide in the Q&A. So this kind of slide summarize the current FDA B guidance versus the M13A in terms of fast and fat B studies. So FDA gonna change from the current recommendation, which is for pretty much for almost all oral IR drug products, asking both fast and fat B studies, will move towards the risk-based approach as delineated in the M13A. And as I already mentioned, so for the high-risk product, probably both fast and fat study, but for non-high-risk product, they could be either fasting-only study or fat-only study, or either fasting or fat. Depends on the situation as delineated in the guidance. So FDA is really leading the harmonization efforts, and ICH recognize the importance of the harmonization in bringing the access to the medicine globally. And because of the M13A not being finalized and adopted, and FDA will revise majority of the IR oral products, PSGs, to align with M13A, following the risk-based approach, by considering the BCS classification of the drug substances, formulation design of the ROD, and what's the food effect, what's the labeling condition, whether the drug is to be taken with food or without regard to food. So based on this assessment, we look at more than 1,100 PSGs, current existing PSG, which covers those products. And we recognize that more than 800 of these TSGs can be determined as non-high risk. So we will recommend from the current 2B studies to 1B studies, either in the fasting conditions or fat conditions, which means we will remove either fat B study or fast B studies based on the labeling condition. And you can see we published the forecast list. There's a link down there last Friday. So you will see what are those 800 plus PSGs are. And we also aim to publish the revised PSG in the first quarter of 2024, which target in October next month. So the significance of M13A is very obvious. So this is the first harmonized ICH guideline focused on BE. It will impact a significant portion of the pharmaceutical market. This is also the reason why the ICH started IR product first, because it's almost occupy about 50% of the market or approved new drugs in the US. And they will reduce the need for additional in vivo BE studies due to the divergent recommendations. And it will support a more streamlined global drug development. Although I did not really cover much, is the M13 also recommend a study design where multiple arms can be utilized in the study design to include the multiple reference products or comparative product or multiple test product in order to improve the efficiency and satisfy the multiple jurisdiction requirement. And this will benefit the patients by increasing access to generic drugs. So here are the key takeaways. So M13A has been finalized, and it's adopted by ICH, and it will be implemented or is in the middle of implementation for ICH regulatory members, including FDA and other regulatory agencies. It will facilitate the regulatory approval, promote confidence in the quality of the generic drugs, and also enhance global access to those medicines. FDA is updating the relevant guidance, not just the PSG, but our general guidance will also be updated to align with M13A, with relate to the IR part. And also just remember, both FDA's current guidance and ICH M13A do offer flexibility. The ANDA applicants may provide appropriate scientific justification if they decide to take an alternative B approach, different from the guidance, deviate from guidance. That is also possible. So today I only can have a very high-level update on this important guidance, but we plan to have a SBI webinar on this guidance on November 21st, save the date, for a 2-hour webinar, which will have additional detailed update on the key scientific thinking behind the recommendations, FDA implementation plan, and address your questions more thoroughly. Now is the challenge question. Challenge question number 1. Following the implementation of ICH M13A guidance, which of the following will represent the most significant changes in the FDA recommendations for B studies for immediate release solid oral dosage drug products? A, B studies design with regard to food conditions. B studies population. C, B for long half-life drugs. D, batch size for pivotal B studies. So the correct answer is A, B study design with regard to food conditions. Challenge question number 2. FDA will revise product-specific guidances for immediate release oral drug products to align with ICH M13A. Is true or false? I hope you get it is true. Thanks. So here are the resources. Hopefully will provide you additional information. And I would like to end that we, as OGD, as FDA, we collaborate globally to resolve scientific and regulatory challenges to ensure access to generic drugs. With that, I'm going to turn over to Yu-Chin, who will provide an update on the MIE program, pilot program. Thanks.
Hi, I'll be providing a talk on the Model Integrated Evidence, the MIE Industry Meeting Pilot Program for Generic Drugs for the first year review. This is my disclaimer. So in today's talk, I'll provide an overview of the Model Integrated Evidence, the MIE Industry Meeting Pilot Program, and share what we have learned through the first year of the MIE Meeting Program, and recognize the potential topics for discussion through the MIE Meeting Pilot Program, and then learn how to prepare an effective MIE meeting request package. So in OGD, there are three major areas to apply the quantitative methods and modeling. They are mechanistic models, including the physiologically based PK, the PBPK, and the CFD models, like we had some talk yesterday and today. This can be applied to both the oral drug and non-oral drugs. And the second main area is the pharmacometrics and the quantitative clinical pharmacology, which includes the population-based model, the pharmacokinetics, and pharmacodynamics models. And the third area is the data analytics, which I put here as big data, including the machine learning toolsets, analytics for complex mixtures, and risk-based model, and etc. OGD launched the MIE pilot program on October 1st, 2023. So the MIE pilot program aims to offer the generic drug company a meeting opportunity who intends to submit an MIE approach in their ANDA. And the MIE program is open for the whole life cycle of the ANDA, including the pre-ANDA and ANDA stages. So the pilot program allows enhanced scientific communications on a broad range of quantitative methods and modeling techniques to address generic drug development issues or questions that are either out of the scope or cannot be sufficiently addressed by the existing pre-ANDA and ANDA scientific meetings. For example, you can discuss the common modeling issues across multiple products and common modeling approaches for non-complex products in the MIE program, which are not the main focus in the current pre-ANDA and ANDA scientific meetings. And the main objective of the MIE program is to provide a dedicated regulatory platform for interactions on MIE to foster early and focused interactions between industry and FDA on MIE approaches for establishing bioequivalence in generic drug development. And you may also refer to the general principle documents link in the bottom. In the MIE meeting, the following scenarios may be granted to discuss the innovative MIE-focused approaches for BE establishment that cannot be effectively addressed under the existing GDUFA scientific meetings, and to discuss the non-complex products with complex modeling approaches, and to discuss the novel data analytics tools and approaches, for example, the machine learning and artificial intelligence for BE establishment and assessment. Here I'm providing a brief overview of the MIE pilot program timeline. So first I'd like to note the MIE pilot meeting is not a GDUFA meeting, so it doesn't follow GDUFA goals. However, we're following approximately the timeline that's listed here. So the MIE pilot meeting includes 3 stages. In stage 1, the applicant submits an MIE meeting request. Within about 14 days, FDA will assess and evaluate the meeting package, and at its sole discretion, FDA will make the decision to grant or deny the meeting. If the meeting is granted, in stage 2, it may include up to 2 meetings with the applicant. So within the 30 days of the grant date, FDA may schedule and host an optional orientation meeting with the applicant where the applicant will present its MIE approach in a video conference and have a direct communication with FDA regarding the details of the MIE package. And whether, regardless if the orientation meeting is scheduled or not, the applicant will have the final meeting with FDA in 120 days to 150 days, depends on the orientation video conference meeting. And FDA will issue the preliminary response 5 days before the meeting days. And the stage 3 is the post-meeting communications. Approximately 30 days after the final meeting, FDA will send the official meeting minutes. Here I'm providing some case examples through the first-year program experience to share with you what is opportunities of this MIE meeting program. So the first case example discussed a complex regulatory issue where modeling and simulation were used to characterize PK differences between available and discontinued products, providing a scientific bridge for BE demonstration under the circumstance where neither the R&D nor the RS is accessible. So in this case, we discussed feasible model-based bridging between the two oral dosage forms, where the applicant will be able to conduct a comparative in vivo PK study for its test product compared with an available product, although this product is not R&D. However, the model-based bridging helped to identify the PK differences between all these products and eventually provide a BE evaluation for the applicant's test product versus the R&D. The MIE meeting provided in-depth technical discussion regarding the data source and the bridging strategy and the modeling techniques. So through this MIE meeting, a feasible model-based bridging approach was established. And I'd like to note that this is a case example where the MIE meeting program allowed to discuss the modeling approaches for non-complex drug. In my second case example, the applicant submitted the MIE meeting request to discuss an alternative MIE-focused approach for BE establishment for a long-acting injectable. So this long-acting injectable, the bioequivalence study needs to be conducted in patient population to reduce the recruiting burden for patients. So the applicant submitted a mechanistic PBPK modeling approach informed by in vitro characterization data and a small-scale in vivo validation study. In this package, here are some example topics that were discussed in the meeting, including the detailed technical discussion regarding modeling methods and modeling validation strategy, and in vitro experimental methodologies for PBPK model development and validation purpose, and the approach for virtual bioequivalence analysis, for example, the selection of the sample size, the power of the study, and the variability, and risk assessment, including type 1 and type 2 error analysis. So since launch, 2 meetings were granted and completed the MIE process. The MIE pilot meeting provided a unique forum for FDA and industry scientists to gain a shared understanding on model development, model performance, and the use of modeling to address a specific issue in generic drug development. So in those meetings, FDA's participation are multidisciplinary, and it provides inputs from the ORS DQMM-led multidisciplinary review team. And the MIE meeting allows alignment in MIE strategy across FDA review team and with the applicant at early stage. And through those meetings, we found an orientation meeting was helpful to understand the proposal at early stages and enhance the mutual understanding. So the video meeting format for both the orientation meeting and the final meeting was especially beneficial as it allows direct communication with the applicant on the complex modeling questions. Here are some examples for the good candidates for the MIE meetings. I'm putting here as 3 big categories, but there could be many, many examples on each categories. So first one is you may discuss innovative MIE-focused approaches for BE establishment to address complex issues for a single product or multiple products in an MIE meeting. Some examples are listed here. For example, like yesterday, we have a talk discussed a common strategy for validating a CFD model or platform towards predicting the regional deposition for oral inhaled drug products. So this common strategy can be discussed under an MIE pilot program. And another example is an alternative study designs for long-acting injectable supported by MIE, as my case example 2 here. You can also discuss the non-complex products with complex modeling approaches in an MIE meeting. For example, in my case number 1, the model-based bridging for alternative product when the R&D and RS is discontinued. And the other example could be the BCS-based bio-waiver or the other study waivers for the oral drugs. You may also discuss the novel data analytic tools in the MIE meeting. For example, the equivalence analysis of complex particle size distribution, the new quantitative approach for API sameness assessment, and application of machine learning and artificial intelligence for bioequivalence establishment. And for more details, you may refer to the SBI 8 webinar in January 2024, where we have a dedicated webinar on the MIE pilot program. To prepare an effective MIE meeting request package, you should consider have a sufficiently developed scientific proposal where the meeting questions are relevant, critical, and clearly stated. And adequate documentation for the modeling approach can be very helpful to be more effective in the communication. For example, the purpose of the MIE approaches and how it will be used to address the question of interest and inform regulatory decision-making, and sufficient details for underlying assumptions and the model building process, and a clear model validation and verification strategy, including how you will be using the data and the future data to support, and risk analysis and assessment, and modeling files and supporting data sets. So you may submit one single request for MIE along with a complete meeting package to FDA at mie@fda.hhs.gov. And a confirmation of meeting submission recipient by FDA will be sent via email that will indicate FDA received date. Here are my challenge questions. Which are the following scenarios may be granted as an MIE pilot meeting? A, innovative MIE-focused approach for BE establishment. B, non-complex products with complex modeling approaches. C, novel data analytic tools and approaches for BE establishment and assessment. D, all of above. So the answer is D, all of above. Challenge question 2, which of the following statement is not true? A, within 14 days of the meeting package recipient date, FDA will strive to evaluate and assess the meeting package submission. FDA, at its sole discretion, will determine if an MIE meeting request will be granted or denied. B, the MIE pilot meeting is part of GDUFA meetings and will follow GDUFA meeting process and timeline. C, during the initial assessment of the MIE meeting package, FDA may determine and schedule and host an optional orientation meeting within the 30 days of the grant decision date. D, regardless if an option orientation meeting is scheduled, the applicant will participate in a final meeting via video conference with FDA. So B is not true, as the MIE meeting is not part of the GDUFA meetings. Here are some take-home message. So the pilot program serves as a dedicated regulatory platform for industry to explore the proposed MIE approaches, obtain FDA's advice on their feasibility and advancement, and address relevant scientific and technical questions. And I'd like to note the MIE meeting program is open for the whole life cycle of the ANDA, including pre-ANDA stage and ANDA stage. And the MIE pilot program is ongoing to accept more meeting requests so that areas of improvement can be identified to support future recommendations. So we highly encourage applicants to participate and engage with the FDA early in your product development program. Here are some resources that might be helpful. The first link is to the general principle guide documents for the pilot program. And the second link is to the January 2024 SBIA workshop, where we had a dedicated webinar to discuss the MIE industry meeting pilot program. And a closing thought, FDA has been openly encouraging the use of quantitative methods and modeling approaches with MIE to support the development and approval of generic drug products. So consider the MIE pilot program when requesting the meeting with FDA. Now I'd like to invite all the other speakers and panelists to join me to the panel. Thank you.
All right, thank you again to our presenters for their excellent presentations, and we will now have our final question and answer panel to include all our presenters, and also joining the panel, we have Dr. Nilofer Tempal, Associate Director for Scientific Quality with the Office of Bioequivalence in the Office of Generic Drugs, as well as as well as Rachel Erdman, JD, Regulatory Counsel with the Division of Policy Development with the Office of Generic Drug Policy within the Office of Generic Drugs as well. All right, and as before, attendees in the room, you are invited up to line up at the mics to ask any questions. And for those online, if you haven't had a chance to submit your questions using the on-screen link and QR code, please do so now, and we'll answer as many questions as time allows. All right. So again, in the room, this is the last chance to ask the experts while you have them here. So at any point, please feel free to come up. And in the time being, we'll go ahead and start with our online questions then.
All right, so our first question is for Dr. Greg Huang. So for lyophilized product, if RLD composition doesn't provide salt form quantity, but only includes free base quantity per vial, as an ANDA holder, do we have to specify the salt form quantity in composition?
Yes, both the salt form and the free base concentration and the name should be included in the composition table. And also it should be clarified in the labeling.
Thank you for that response. And now we have a question in room.
So the question is for Dr. Huang as well. So is excipient function part of the Q1, Q2 evaluation? So for example, if we match in all the other chemical characteristics as well as the quantity, however, we list the different primary function as the RLD holder, would that make us not Q1 equivalent?
This is an assessment question. Basically, the terminology may be some different in the naming, but it should not affect the Q1, Q2. Q1, Q2 is based on the qualitative and quantitative information, but we understand that some different applicants may use slightly different naming, but the role should be equivalent. And
for example, like BHT, we list as antioxidant, they list as preservative. But this is an extreme case since they're both exceptional. But in that situation, they're quite different in terms of their functions. Would that be okay?
Yeah, you might get a question if we are not clear about your description of the role of the excipient. Yeah, you might get a question in the CMC review.
Okay.
Thank you.
Thank you for that response. We'll go to another online question.
All right, so we have another follow-up question for Dr. Huang. Should all inactive ingredients be included in the outer pack or container label?
Yeah, all the ingredients should be included in the labeling. The name, the list, the full list.
Thank you for clarifying that. We'll go for another online question.
All right, we have another question here for Rachel Erdman. So should applicants round the amounts in the statement of ingredients in the labeling?
Well, thank you. Generally, no, we don't request that you round the numbers. The numbers, significant figures should match what's in your composition statement. Thanks.
Thank you for that response too. We'll go ahead and continue with our online questions.
All right, this question is for Dr. Fong Lu. If OPQ identifies GMP issues related to an API supplier, but the applicant showed that the API batches do not have concerns related to GMP issues, will FDA accept the biostudy?
It's case-by-case scenario. Semi-justification we can consider.
Thank you. And we still don't have any takers in the room quite yet. Oh, okay, actually we do. All right. So thank you for coming up, and please go ahead, ask your question.
This was a great program. This is a hypothetical question to anybody in the panel or for the entire thing over here. I've always seen that, okay, brand drugs, and now we are talking about generics. So in case the brand manufacturer decides to make the first generic, does it mean that he has to just do a label swap? Like, does he have to do an ANDA with just a labeling requirement, or what happens there? You know, if it's the brand, like, let's say you can take, I don't know if it's Mylan or Teva, for the EpiPen, they made the generic epinephrine injection, right? Was there an ANDA process for that, or was it just, like any of the products, like let's say any of the monoclonal antibodies or any of these really complex drugs, like do you have to do another ANDA process even though it's the same manufacturer who's got the brand approved?
Okay, so I'll try to.
Kind of, that's why I didn't want to ask anything, but it's the question that's been intriguing me the whole 2 days, you know, like, because there is a commercial value in having a generic product because you don't want to lose the market. So if you decide to, because it's going to expire, it's going to, so I don't know what's the FDA take on that.
Okay, so what you are referring to is really the authorized generics. That's what we call as the authorized generics. So you, as the innovator, when you want to market this as an authorized generic, all you're doing is just changing the labeling in the front. Then there is no ANDA submission. There is not that I'm aware. I wouldn't say there is no ANDA submission for that. But if you are marketing an authorized generic, you have to, I'm not an expert here in this authorized generic field, but in terms of you have to inform or there is a authorized generic site, you get an NDC number and those things. So you may have to just look into the FDA's, some website for unauthorized generics for additional information. Thank you.
All right, what a great example. If anyone else here has some questions they've been wondering about, now's a great time. We'll do another question here, actually, yes?
Hello, yeah, I have a similar type of question. So, some of these drugs which have been approved long back, ages back, now as you get a new drug or a generic drug product out, you go through a full characterization, and there's a whole bunch of specifications, including impurities and stuff like that. And as part of the ICH guidance, with the impurities, you sort of show some of those things. What if the brand themselves have those impurities which are higher than that, the ICH guidance? What happens in those cases? And it was not in the specification because it's approved ages back.
I think maybe this is a little out of scope of this particular session, and I'm not sure if anybody else wants to answer, but I think this is a little out of scope on this ICH and impurities.
Sure. All right. Well, thank you, Sil, for being willing to ask and that response. And actually, we'll move on to an online question first. Yep.
All right, this question is directed towards Dr. Priyanka Ghosh. The first thing they said was, thank you for providing a very informative presentation. Regarding the characterization-based BE approach, emphasis on the IVPT and IVRT aspect for establishing the BE of topical products, Will there be a bioequivalence data summary table that is formatted and tailored for this type of submission, as the current model bioequivalence data summary tables seem to gear towards clinical data? Thank you.
So at the moment, we do have some summary tables available for IVPT and IVRT that are accessible through the FDA's website. However, if you run into challenges as you are trying to utilize them, I would encourage you to let us know so we understand where the challenges exist and we can update them so they are easier to use.
Thank you for that response. And we had another in-house question.
Hi, thank you for coming out today. My question is about the ICH M13 and harmonization. So first, we thank the FDA for driving and leading some of that harmonization, all those efforts. We understand the challenges of that. Where I work at AAM, we work with one of our partners, IGBA, the International Generics and Biosimilar Association, and we advocate on certain issues. One of the issues we talk about is single global development. And with that, we think about three pillars. We think about the legal frameworks in different jurisdictions. We think about harmonization of BE, which you talked about. We also think about single global development or single global comparator. On slide 76, you mentioned multiple comparator, multiple regions. Can you give us some thoughts or some ideas of how we move the concept of single global comparator down the line, or how we push that concept a little further, so we're not doing multiple comparators in multiple regions for the same product? I hope that's not out of scope.
Okay, I will get a start, and Lufa can always add on. Yeah, we heard you. We did hear that feedback. But however, in the guidance, we did talk about the selection or acceptance of the comparator product. It is really bound by the regional legal jurisdiction. So it's like ICH will not be able to harmonize that in terms of, which acceptance comparator will be accepted by which regulatory agency. So the solution we provide is really what you mentioned is you could, in a single trial, you can include more than one comparator. And if you only rely on one for one, so the multiplicity, we also talk about multiplicity adjustment. In that case, you don't really need to adjust the multiplicity if you only plan to submit the set of data to one particular region for the one particular comparator. I don't know if that addressed your question, but that has been discussed, and we just don't have the bandwidth, I think, in ICH to harmonize that. Do you have any?
Yeah, it's more than the bandwidth. I would say that's just a guiding principle of ICH, that we harmonize on the whatever standards, on the scientific and technical standards of whatever the expert working group is working on, while staying within the jurisdictions, or the regulatory framework of the different regions. So since every region, like EMA, USA, each one requires a very specific reference standard against which the study needs to be done. That's something which is dictated by the regulations, and so that's why it's out of the scope of ICH. So yeah, it's not ICH, maybe some other way to do it.
Please also consider comparator variability from scientific perspective.
All right, thank you all for that. We'll go to one more in-room question here.
I have a very simple question. Does the MIE pilot program cover only generic drug or new drug as well, or maybe drug device combination as well?
So I will start, and they can add. The MIE pilot program currently covers generic drug. You're asking for drug device?
New drug and the drug device combination.
So in new drug, there is an MIDD program, which has similar function to discuss the modeling. So for the MIE, it's the one I'm showing, it's mainly focused on generic drug.
Thank you for that clarification. Now we'll do some online questions.
All right, so our next question is for Dr. Lei Zhang. So prior to the M13A, I was instructed by FDA to conduct both fasting and fed studies for my product, which I think is non-high risk based on M13A. Now M13A is under implementation. Do I need to submit a controlled correspondence asking the FDA to adjust the recommendation, or will it be sufficient to rely on the revised guidance and move?
Yeah, thank you for this question. Yeah, now we are in the middle of revising all the product-specific guidance. I would suggest look at the forecast list. If it is being listed as a being in the plan for revising, that's a hint about what direction FDA's current tentative thinking is. But you can also wait till the PSG is published. You can follow that. I do not think the person need to submit another control correspondences, but if they choose to, it's okay. But I don't think it's really necessary. They should follow the FDA guidance. Thank you. Updated guidance.
Any more comments from the panel? No? Okay, we'll move on to the next online question.
All right, this question is for Dr. Nilofer Tempal. So with this, what should a firm do if it is about to conduct studies under both fasting and fed studies based on previous PSG recommendations for a non-high-risk product? Should it obtain permission from the FDA to drop the fed study, or should it just do it on its own?
No, if you are planning to do 2 studies and if the revised guidance is already posted, which is recommending maybe 1 study, then that guidance is what you should generally be following. So is that the question? I'm sorry.
I believe so, yes.
Yes, okay. Yes, thank you.
All right. So we'll go ahead and do another online question.
Excellent. This question is directed to Dr. Feng Liu. So if applicant uses alternate API source because of CMC issues with the original API supplier and submits CMC data along with the comparative dissolution data during a pending ANDA, will it suffice requirements for ANDA approval, or does the applicant have to conduct BE studies with alternate API source batches?
Can you repeat this question?
Of course. If an applicant uses alternate API sources because of CMC issues with the original API supplier and submits CMC data along with the comparative dissolution data during a pending ANDA, will it suffice requirements for ANDA approval, or does applicant have to conduct BE study with alternate API source batches?
First, to figure out if the API source or supplier has any CGMP issue, I will refer you ask OPQ.
Yeah, we would suggest that you ask this in an appropriate response to FDA controls correspondence, since this is very application specific. Thank you.
Yeah, thank you for clarifying that. All right, and again, we'll continue with our online questions, but still in the meantime, if anyone in-house has anything, feel free to come up. The invitation is still open to come to those mics. So we'll go ahead online.
All right, so this question is for Dr. Greg Wong. So if we have complex inactive in formula, is it okay to list only flavor name, or should we include all components of that flavor?
All components of the flavor and the quantitative information should be provided in module 3P1 composition. Okay, but in the labeling, you may not need to provide that detail about the flavoring agent or coloring agent.
All right. Next question from online.
All right, so this one is for Dr. Yuchen Gong. Regarding MIE meetings, can we in industry use that to discuss modeling certain aspects of device design for a drug-device combination product? We invest a lot of resources into comparing these products, and we could use a modeling approach, including using virtual study subjects for comparative use human factors, for example.
Yes, the answer is yes. You can discuss those type of questions in the MIE.
Thank you for clarifying that. And so we'll keep on going with our online questions.
All right, so this question is for Priyanka. While I understand your presentation was limited to topical products and their respective draft guidances, they, however, list other dosage units that are not within the scope of these guidance documents. When working with IVRT and/or IVPT studies for these other dosage forms, for example, transdermal systems, is it recommended to apply as much as possible within these guidances to the IVPT study design, or are there additional guidances for these dosage forms expected in the future?
Okay. Yes, my presentation was focused on topical products applied to the skin, where we do recommend IVPT and IVRT studies within the scope of characterization-based PE approaches. When it comes to IVPT, and since transdermal dosage forms were mentioned in the question, I think we already do have a guidance, the product quality guidance for transdermal products, that has recommendations related to how IVPT studies can be conducted, for example, to support evaluation of heat effect of a generic transdermal product compared to the reference listed drug. In other situations, I think we currently do not recommend IVPT studies specifically to support product approval. So if you are wanting to conduct such studies, then I would encourage you to chat with us using the pre-NDA pathway.
Thank you for that response. We'll go ahead and go for another online question.
All right, this question is directed towards Rachel Erdman. FDA recommends listing the established name of an inactive ingredient in the statement of ingredients in a drug product's labeling. What if there is no official designated name under Section 508 of the FD&C Act?
So if you're saying that there's no official name and there's nothing in a compendium or anything like that, like USP, then you can generally use the common the common name of the drug, and of course just double check to make sure that that's acceptable. Thanks.
Thank you. Do we still have some more questions coming in online?
I can get some.
Okay.
So we have this next one is directed towards Dr. Fong Liu here. Can we consider the dissolution data from the comparative dissolution testing supporting the API source change as the same as that of in vitro dissolution testing for supporting waiver of in vitro BE study requirements for the lower strengths?
No, it's different. Number one, let me answer. Number one, for waiver of your lower strain, the study should be conducted with. Let me figure out. It's different with T versus R. For the other situation, like for API source change, this should the in vitro dissolution conducted with pre-change with post-change of T. Those different is T versus R, pre-change T, post-change T. That's different.
Thank you for that. All right, and let's see, do we have, okay. Any other last call for questions here in the room? Again, this is the last chance, don't miss the opportunity. All right, well, in that case, we'll go ahead and thank our presenters again and panelists for all their helpful information. And give them another round of applause. Okay. And panelists, you can go ahead and take your seats, but everyone else, if you want to stay put just for something special, and that is our closing remarks from Dr. Robert Leinberger, who serves as the Director of the Office of Research and Standards, or ORS, in the Office of Generic Drugs, or OGD. Dr. Leinberger leads OGD's implementation of the GDUFA science and research commitments, including internal research activities and external research grants and collaborations to ensure the therapeutic equivalence of generic drug products. And ORS also provides pre-submission advice on complex generics through pre-ANDA meetings, product-specific guidances, and correspondence responses. So I'll hand it it over now to Dr. Leinberger.
Thank you very much. I'm very excited to be here to close out this meeting. We had a fantastic workshop. Hopefully you all learned a lot about the science behind the generic program and some of the ways that it makes an extremely positive impact on our review processes, on the development of your products that you submit to FDA, and on access to generic versions for the American public. The generic program won't exist, especially for complex generics, without a strong scientific foundation. It's, if you didn't have that, then it's not clear, well, what studies do you have to do? You'd fall back to a situation where let's just repeat the studies that brand products did. And, that pathway is not going to lead to efficient access to generic products. And I think you saw throughout this workshop the way that the scientific foundations of the generic program are being built up and lead to more efficient product access. On the first day, you saw looking at in vitro release methods for complex suspensions and the scientific work that helps you develop those approaches. You heard about topical products and the Q3 approaches and how they're taking over from the clinical endpoint studies and being the more appropriate pathway for products. You heard a session on inhalation products. Again, not very many generics for the inhalation products, but you saw the progress toward inhalation products based on PK and in vitro studies instead of clinical endpoint studies. We hope this will lead to a surge in interest in generic companies in developing inhalation products that meet the needs of the American public. Again, a huge area of opportunity for you in the generic industry created by our scientific work. And then you also heard about drug device combination products and some of the challenges in developing those types of complex generics. We don't have access to generic products unless you all in the industry submit applications. And I think you heard today in the early morning talks, a lot of examples of some of the things that go on during the review process for some of these complex ANDA products. And hopefully this gives you the insight as to how to make your submissions better, but also that you ought to be investing in development programs for these complex generics because you see that it's possible. You heard about long-acting injectables. I think 10 years ago, can I make a generic for that? It seems impossible. Now it's not just possible, it's done. And if you're not doing it, you're probably falling behind. Same thing with, you heard a talk on peptides, right? 10 years ago, oh, this is a recombinant source RLD. Generic's not possible. The only thing I can do is a B2 application. That's not true anymore. Again, another huge opportunity just in time for what we know is now the biggest part of the pharmaceutical market for generic competition to be active in that. If we didn't build the scientific foundations to change how we're thinking, then we wouldn't have the possibility of generic versions for all the GLP-1 agonists. So again, huge foundation. If you're in the generic industry, keeping up with these are how you plan and establish your business to be competitive in a new environment and provide the things that the American public wants. They want access to generic versions of all of these complex products. And the bigger the impact the products are, the more that they want and need those generic products. And this is, the huge value that you in the generic industry create. We at FDA recognize that, and we constantly try to develop processes, procedures, way to interact with us to make that process more efficient. You heard about our model-informed evidence meeting pilot, right, as well as other ways you can interact with us. And finally, today you heard about the M13 updates. So the scientific force toward more efficient bioequivalence, not just limited to complex products. Even the commodity tablets and capsules, we continue to try to make that process much more efficient. And I think, look at what the impact of that is. Look at that forecast list that Lei mentioned. 800 PSGs tells you what study is going to be removed. I would think you're going to get a lot of questions about that, but this is, again, a huge restructuring. If you think about the benefit of that, that's probably 400 Fed BE studies that last year you would have submitted, next year you don't have to. That's, I don't know how much the studies, I know how much PK studies cost me, I don't know how much they cost you, but that's hundreds of millions of dollars in savings for the generic industry. And that's not just savings next year, right? That's savings sort of every year going forward that you're producing and maintaining these products that are essential to just the everyday use. I mean, they're commodity products, but commodity product is still making a huge public health impact. People take commodity products every day for blood pressure medications, cholesterol-lowering medications, huge public health impacts from high-quality commodity products, and we continue to make that process easier and more straightforward to make those products and ensure the supply of that. So we hope that you take that money that you save on those BE studies and you can invest it into product quality, reliability, and ensuring continued access to these products. And with that, this is a fantastic opportunity for you all in the industry, in the room and online, to get in touch and hear about these innovative approaches and what's changing, so you can keep ahead of the, all of these things are going to have significant impacts on your businesses, business processes, and scientific developments going forward. This is a fantastic place to keep up with that. And I do want to thank the people that really made this possible, especially the scientific organizing committee. So we have one, I don't think Wen Lei is here. I want to recognize her as the chair of our scientific committee. I want to recognize Lei as our executive sponsor to keep everything working, and also recognize Maria as leading the project management activities on that. But we also had a large committee of scientific experts. You saw many of them as the panelists or the leads session moderators for today. Just recognizing a few of them, recognizing some of our project managers, Irfan and Karen, recognizing Jan, Andrew Babiskin, Deanna Vivian, Key Ren, Susan Levine, Fallon Smalls, Sheila Rajesh, Ahmed Zidan, all of these people from OGD and OPQ who are members of our technical committee who really helped put together the programming, recruit the speakers, organize the topics, help develop, moderate the questions. So we had a great discussion. So we want to thank all of those people. So join me in thanking them. And with that, I'll turn it over to our SBIA staff to close out the meeting and tell you how to get all the final benefits from this meeting and everything you need.
All right, thank you again, Dr. Leinberger. So now here we are, all ready to wrap up Advancing Generic Drug Development Workshop 2024. Just a few final reminders as we close out our day. Recordings will eventually be posted on our FDA.gov CDER SBIA website, but in the meantime, you should be able to access the YouTube streaming links on our One Stop Shop webpage. Please be sure to use that evaluation link to obtain a certificate of attendance to support continuing education credits from SOCRA, RAPS, SQA, and ACRP. And also note that the CE claim codes will be sent to attendees for continuing education credits for healthcare professionals, including physicians, pharmacists, and nurses. But please note the codes will be emailed within 24 hours of the conclusion of this event. And again, that evaluation and certificate is available for 2 weeks only. So that's going to be until October 9th, 2024. And one additional reminder for in-person attendees here that have stuck it out with us all the way to the end of the day here, we want to make sure that you get your parking discount. So if you haven't already gotten that, to be sure you only pay $10 for the day, please stop by the registration area and we'll make sure you get that covered. All right, so thank you once more to all who joined us for this 2-day workshop and made it a success. This concludes today's sessions, and we look forward to you joining us again for future events.